customs, I said, well, if I can get through customs...
I remember your
Yeah?
comment. Yeah, I remember your comment.
Which was funny.
But really, I haven't understood that. So I was like...
I'm very glad you printed it. So look, shall we... Yeah, I also grab a pen. Shall we alternate?
Do you have a pen?
I'm going to get... Shall we take turns?
Yeah,
on
we
this?
alternate, yeah. That was a little bit outside Lisbon, in Sintra. And I thought, no, I should not go. But yesterday, my colleague that was going to substitute me told me, well, João, but if you have the session at 1:30...
You can easily make it.
But I decided not to go.
I think many people will expect it to be finished starting at 1:30, so they will creep in through and miss the first guy.
But we have to start on time, right?
Who is... Oh, he was there.
Where is Raquel?
Here. This is Ellie Nadaf.
Ellie Nadaf, so is the first one?
Neurology, yeah.
So
Olivier Boyer we know, but I haven't seen. I imagine he is here.
But he is the third, right?
He's second. You start with him.
Okay.
Olivier, they've spelt him wrong, but anyway, doesn't
Is
matter.
he the second?
Yeah.
You remember the program, is it?
Well, let's check.
Because, you know, Katerina told us that she was the second. We have to check that.
We had another email.
No.
Okay, I might have the app. Good God, this is the problem of having nothing printed, you know? It's not helpful. You have the app?
No, I have the program here.
Oh, well done.
Have you downloaded the app?
Excuse me 1 minute now.
Hello? Ah, Raquel, olá. Estás boa? So, but... Olá, Eduardo. Olá, Eduardo. Tudo bem? Nós recebemos... Eu hoje de manhã enviei um mail ao Federico a pedir as afiliações... Olá. As afiliações deles, é isso, não é? É. Exato. Eu recebi. Não tinha, eu é que pedi. Pronto, mas está tudo ok. É só a ordem dos nomes é que não coincidia com a ordem do programa. 1, 2, 3. aqui Olha, Raquel, vamos começar a horas, não é? Yeah. So we'll start now.
3, 4. So why don't you open and...
Okay.
Olivier, bonjour.
Yeah, 3.
It's okay.
Hello.
He knows what's going on. Over to you.
Okay. I'll manage the start.
So we have Olivier. I haven't seen Thomas yet, but I'm sure he's around.
He should be around. So, good afternoon. Be very welcome to Lisbon. Be very welcome to the 6th Global Conference on Myositis. It is a privilege from two of us to open, in fact, this conference. We will have a few talks and also some brief communications from the abstracts. I hope that you're feeling okay. There's a nice weather outside, and so things are all set to have a marvelous conference, but also a marvelous day, and the following will be also as this one in Lisbon. So I hope that you enjoy the period of staying here in Lisbon. I'm also from Lisbon. My name is João Fonseca. I'm professor of rheumatology here in the University of Lisbon. And on my right hand is Lucy Vanderom. She works in London, and she's much more known than me in the world of myositis. And I would like to call for the first talk, Eli Nadaf. Dr. Eli Nadaf is an associate professor of neurology, dedicated himself to neuromuscular medicine and muscle pathology at Mayo Clinic in Rochester. His research focuses on inclusion body myositis and the intersection of mitochondria function, inflammation, and aging. And he also serves as the institutional leader for CAR T-cell therapy for non-cancer indication at Mayo Clinic, Rochester. So please.
Good afternoon, everyone. Thank you first for the invitation to give this talk that's dear to my heart, as that's an area I enjoy researching. It's about the role of mitochondria in the pathophysiology of idiopathic inflammatory myopathies. As non-financial disclosures, I'm a clinician scientist. I'm not a mitochondria biologist. So I'll just mainly present some features to you that I'm familiar with about mitochondrial biology. It will be a lot to cover in one talk, so we will just be mainly doing some illustrative examples from pathways that are relevant to mitochondria. First, we're gonna do a small exercise together. So I want you to just look at this photo and judge the person that's in the photo, the one in the white, not the one in blue. And I'll tell you a little bit more about them and see if you can tell who he is, what he does in life, what's his role, and so on and so forth. As far as activity, he loves sauna. He spends most of the time at the gym in the sauna. He travels for fasting or starvation workshops. And so, yeah, you can judge him. He's one of those that want to do anything but exercise. But the reason why his photo is there, so one day in the sauna, he asked me about mitochondria. And I started telling him, and he said, wait, Eli, you have to choose one test to do, and that test will tell you, is there mitochondrial dysfunction, yes or no? So what would that test be? And I didn't know at that time, and I still don't know. But as you can tell, he's a neurologist, so he's also a neurology colleague, and you could probably guess by his question, simple, fast-paced question, that he's a stroke neurologist, and you'd be right. But you can't tell a lot about him just by looking at this photo. You have to observe him. in different situations. You have to observe activities he does, function he does, and even so behaviors to tell who he is. And not just while relaxing, like eating a pastéis de nata on a miradura, you have to see him under stress. And there is a healthy amount of stress that builds resilience, like when we exercise. And there is an amount of stress that we cannot overcome where our mechanism of defense fail down, and that's what happened in diseases. So same thing applies to mitochondria. That traditional historical concept of mitochondrial dysfunction seems not to be true. Mitochondria doesn't just produce energy. It has so many roles. It's the central hub of metabolism, and most importantly, it's a signaling organelle. Is there a pointer or no? It is a signaling organelle. That means it communicates with the nucleus. It tells the cell what to do, the immune system what to do. It helps at least contribute to determining the cell fate, and so on and so forth. So there's nothing as a mitochondrial function. There are a series of mitochondrial functions and behaviors. So there is no disease, I think, that has mitochondrial dysfunction versus not. You could argue anything that happened to the muscle, even not a disease, the mitochondria will be involved. The question is what type of mitochondrial involvement is occurring. And you could just look at it from a feature perspective, like you just looked at the photo of my friend. That's more like RNA and protein. And you know a bit more from activities, like complex activities, even more if you look at functions or behaviors such as signaling. So we owe most what we know about, you could argue, myositis pathophysiology to detailed description of histopathological findings. And as far as mitochondria, there are three main stains there. That's the trichrome, modified trichrome. I'm showing the red fibers with accumulation of subsarcolemmal mitochondria. It's about the same on succinate dehydrogenase complex 2 stain. Complex 2 is pretty well preserved complex. And complex 4, which is a bit more sensitive to diseases and aging, you see here the cytochrome C oxidase negative fibers. And we knew for decades that there are mitochondrial histologic abnormalities in myositis, especially in IBM, and way more than expected with aging. And there is also, we know that mitochondria have some abnormal feature based also on detailed electron microscopy descriptions in various types of IAM, but more so in IBM. So the first step, a general assessment of a mitochondrial involvement in tissue, is to look at the mitochondrial content, because you could probably see that this tissue here has more mitochondria than this one, so it will be breathing differently and behaving differently than the tissue with lower mitochondrial content. The challenge there, you cannot directly measure mitochondrial content in frozen tissue or non-live tissue. So there are surrogate measures for that. Probably one of the most commonly used include mitochondrial DNA content, citrate synthase activity, or measurement of OXPHOS complexes. But as you can see, these can also be involved in disease pathology. So they could over or underestimate your content. And an example from myositis, so there is evidence of low mitochondrial DNA content, especially in IBM. Sorry, I don't see my pointer on my screen, so I'm pointing at the wrong place. So there's increased mitochondrial DNA deletions and decreased content in IBM, probably also in other IAM, although the studies there are more limited with the smaller numbers. So we're going to approach it as with all these features and behaviors and activities. We're just going to do a hypothesis-driven approach. So you cannot talk about myositis without talking about inflammation. And then you cannot talk about inflammation without talking about mitochondria. Traditionally, we talk about oxidative stress damaging the mitochondria. But now, as we know more and more, it's actually the mitochondrial role is way more important. Mitochondria can start inflammation, activate it, and also is important to turn it down, as we'll see with some examples. And there are different features there that would be too much to cover in 25 minutes, so I'm just going to focus on one part. But for you who are interested, I would suggest also one talking about immunometabolism and how the switches in metabolism determine cell fates, for example, which we will not cover here. We're going to cover one part of it, which is the activation of the immune system by the mitochondrial content. Why is that? Because as you know, life was made possible by a symbiotic relationship between a bacterium, which became the mitochondria, and a eukaryotic cell. So as the reason why this relationship has been successful, because there have been mutual respect and everyone has their own content. So the mitochondrial content, you can imagine, has been isolated from the cell for billions of years. But the bad habit of mitochondria, when they get damaged or sick, they leak. And when that content gets exposed to outside, it triggers major inflammatory responses. And many of these have been very well detailed. Later on, you're going to talk about interferon. For instance, cGAS-STING pathway is a major trigger for interferon-1 responses. Patients with primary mitochondrial myopathies, a non-immune disease, have increased expression of interferon-associated gene. But I want to highlight today a part of the immune system not often talked about. It's the innate immune system, as we're more focused on B and T cell. We're realizing more and more the major role of the innate immune system, especially in cell fate. For example, these are the inflammasomes that are macrocomplexes that usually, if they are activated, triggered and activated, they oligomerize in forming a macrocomplex here, the NLRP3 inflammasome, that result in activation of caspases and secretion of interleukin. And one of the main roles is actually to execute the cell. So there is a level of damage. If you go beyond, it's healthier for the tissue to just execute that cell to restore homeostasis. And it's now a hot topic in neurodegenerative diseases, neuromuscular diseases, and so on and so forth. There are so many pathways to look at, so how do we prioritize? As a translational physician, I think there are two requirements for a pathway to be relevant, at least to the clinical part. One, they have to have clinical significance, so they have to at least be associated with certain clinical measures. And two, we would also prioritize pathway that there are drugs at least later in the pipeline to target. Is that the pathway relevant? So this is here an unsupervised in IBM approach. It's just bulk RNA-seq basically from 60 patient IBM and control. And you can see the top upregulated pathway is the NLRP3 inflammasome that's increased in RNA, protein level, and so on and so forth. And that's also seen in different studies. Actually, I looked when I worked on that project, every single RNA published paper, you always find it there in the top 10, whether called NLRP inflammasome, whether it's called neuroinflammation, and so on and so forth. And even with more targeted analysis here by Dr. Schmidt and his group that reported highest expression of NLRP in IBM, and some also earlier paper reporting increased level in DM. What could trigger that inflammasome, if we can go back there? One of the main content that trigger inflammation is mitochondrial DNA, and there are others also, mitochondrial molecule. But mitochondrial DNA is a known strong, it really triggers the inflammasome and other inflammatory pathways. And for instance, in IBM, with Dr. Kleeffeld's group, there is increased amount of circulating cell-free mitochondrial DNA there. Less studies are done in other myositis, I would say. So now going to the different features of mitochondria. So now here, for instance, we're seeing what correlate with the NLRP expression on RNA level. And you can see a lot of the positive correlation to the left are related to the immune system. A lot of complement pathways correlate with NLRP, which remain a little bit underexplored in IBM. But when you go to the inversely correlated, you see a clear mitochondrial signature there. Why? Because we talked about how mitochondria trigger inflammation. Now we're going to see why mitophagy is one of the most important features to actually turn down inflammation and maintain mitochondrial health, or the term that I like, mitochondrial fitness. Because usually, under normal condition, damaged mitochondria get eliminated by mitophagy, so you can have new healthy mitochondria that come in and you maintain a healthy pool of mitochondria. And how that can play a role in tissue, for instance, under normal circumstances or under healthy level of stress, you have a stressed mitochondria, it leaked, it triggered the inflammasome, then mitophagy kicks in, eliminate those damaged mitochondria, and you get your homeostasis restored. In disease, under disease states, for instance, in IBM, where you have also alteration defect in mitophagy, now the mitochondria leaked, triggered the inflammasome, you have defective mitophagy, so you have more accumulation of damaged mitochondria, and then you have more a positive feed-forward loop that just becomes like a vicious circle that results in cell death. And the main mitophagy pathway that to look at is ubiquitin mitophagy. There is the receptor-mediated mitophagy, and the canonical mitophagy, if you wish, is ubiquitin mitophagy. Very briefly there, so when the mitochondria is under stress and there is alteration in membrane potential, PINK1 stabilizes on the membrane, it phosphorylates ubiquitin at a serine residue, serine S65, and that activates Parkin. And Parkin is a ligase. It just tags all these protein with ubiquitin. Now your system, your autophagic system, see these mitochondria are tagged and needs to be eliminated. So PS65 ubiquitin, I'll call it phosphoubiquitin from now on, is a very, very well validated and dynamic marker of mitophagy. And we have very limited studies in myositis, in muscle diseases in general, but at least in IBM, we can see marked elevation of phosphoubiquitin levels, indicating basically there is accumulated damaged mitochondria, more significantly in males. And when you look at histopathology, there are here phosphoubiquitin aggregates in some muscle fibers. Interestingly, all these muscle fibers are type 2 fibers. And having someone who've read many IBM biopsies, I've never paid attention until then that actually all CCO negative fibers in IBM are type 2. And type 2 fibers are known to be more prone for metabolic disturbances and mitochondrial dysfunction. When we looked at a bigger sample size, I think it's altered in both in males and female, but still much stronger in males. And as we talked about clinical correlation, the levels strongly correlate with disease severity. Here's MMT Mayo scale, so the higher the score, the more severe the weakness. And you have strong correlation between higher damaged mitochondria, more severe weakness, especially in males. So that's just a bit overview of mitochondria role in inflammation. Now, shifting gears, we'll go back to the mitochondria powerhouse of the cell. It's still one of the major, if not the main function of mitochondria is actually to produce energy. And here where that pyramid plays a role, because you could just look at features, RNA and protein, but the more you want to look in depth at mitochondrial respiration, you have to really go up that ladder and look at the respiration itself. However, this is very challenging because once you freeze tissue, there are no reliable methods to do that. And mitochondrial respiration is the main index of mitochondrial fitness. So going back to the question, is there one test to tell you if there is something wrong or not? Well, if you want to choose one, then probably mitochondrial respiration and mitochondrial spare respiratory capacity. As a brief reminder here, electron goes in through complex 1, feeding on NADH, and complex 2. Then they get transported, which creates a proton gradient, and that when it goes through complex 5, it helps with, or it creates ATP. And there are different protocols there on how you could look at that, so the high-resolution respirometry. So basically how it's done, you take a piece of muscle and you dissect it under the microscope until you have single fibers. Then you can permeabilize them, and then you put them in the Ouroboros machine, and then you start feeding substrates and blockers, and that's your protocol. So basically here, when you put substrate only, you're looking, so you always have high oxygen in the protocol we use, so you're not limited by oxygen. You give the mitochondria first the substrate that it needs without ADP, and that's like your original substrate only, which is a leak state. Then you give the mitochondria high ADP, so now the mitochondria in state 3 has all what it needs to breathe. So the only limitation is its OXPHOS capacity. Then you can block complex 1 to see how much is coming from complex 2, then block ATP synthase, leak state, and at the end, you just permeabilize the membrane where you're uncoupling, so electrons are just free-floating, and your main limitation is electron transport system capacity. So we did that in IBM. That's now posted in a preprint on bioRxiv. We did a large number of IBM and control. And you can see in all maximal states, there is lower respiration per tissue in IBM. So the tissue is breathing less, the mitochondria are breathing less in IBM, both in males and female. And you'd wonder why. Well, one possibility is you just have, everything is great, you just have lower mitochondrial content. So in that case, we measured citrate synthase activity, and we normalized for citrate synthase activity. And you can see, to some extent, it does correct with that. So there is some contribution with lower mitochondrial content there. But the interesting thing with mitochondria, it's very, very sex-dependent. So to really see what's happening, you have to segregate by sex. And now if you look at males, yes. So when we say we normalize to citrate synthase, you're almost like evaluating the mitochondrion itself, how much it's breathing, and there is decreased maximal respiration in males, while females have higher leak. So ideally, you want to have maximal respiration with the lowest leak. So that's why you can use these ratios that are independent of your content or tissue weight, and you compare how you're breathing, maximal breathing, to your leak state. And that's your respiratory capacity ratio that's markedly decreased in IBM with higher leak ratio in both male and females, although the underlying mechanism are probably different. Is that of clinical significance, where there's a very strong association, especially in male in IBM, with mitochondrial respiration with age, with the aging component. And interestingly, in female, the more severe the diseases, the longer the duration is, they switch breathing from complex 1 to complex 2. And one thing with mitochondrial involvement, you cannot assume its impairment. It could also be an adaptation. So everything you observe, it could be detrimental, or it could be some protective mechanism. For example, if you breathe less from complex 1, you'll have less reactive oxygen species than complex 2, and you stimulate more biogenesis to make up for that low mitochondrial content. You could measure reactive oxygen species simultaneously, the maximal emitting capacity of frost during each state, and you look more at the leak state, which is where you see the highest level. And unexpectedly, I was thinking, well, it's myositis. We should have high ROS emitting capacity in IBM, but that actually lower. First, I thought there's something wrong. We validated, repeated. Meanwhile, there is another risk, because there are no studies in muscle diseases. Then there's another study in myotonic dystrophy type 1 just got published, and they found the same thing. Then it makes sense. Well, maybe because you're exposed to that ongoing amount of stress, you build a better oxidative, antioxidative capacity there, which is actually was true. Just a crude evaluation of total antioxidant capacity that was higher in IBM, especially in female, where it strongly correlates with disease duration. Last part, we'll talk about metabolism, because you cannot talk about myositis, you cannot talk about mitochondria without talking about metabolism. The thing with metabolic pathway, you could say they are just not too simple to deal with and not easy to, you could at least summarize them in one slide, but they're not like RNA, and there's a great review article about that, how the metabolites should not be treated like RNA. It's not like if you have two metabolites, then that pathway is upregulated. You really have to zoom out. And in muscle, it's very important because there are so many factors that determine the muscle metabolism. And that's a source of sometimes of frustration and disconnect with the patients, where their muscle is great and their strength is great, they just get tired and they have problem with endurance. It's in part due to the immune system, but also mitochondrial health, cardiorespiratory health, all the drugs we use to treat, and so on and so forth. I just want to show you one quick example here. So there is the untargeted metabolomics where you get more fractionated picture, but it's good to generate some hypotheses. Or if you know where you want to look, you could just go with direct measurement. But there is an in-between novel platform called central carbon metabolism that's semi-quantitative and focused on the mitochondria. For the sake of time, I'm just going to show you the zoom-out picture in IBM. That's where we did it, that show a shift from glycolysis to pentose phosphate, and that's the main generator of NADPH and precursor for nucleotide synthesis, with anaplerotic increase in all amino acids that are feeding into the TCA cycle, which now people are wondering about actually hypermetabolism driving the muscle to melt down and fail rather than just failure of energy. Last but not least, sex differences in mitochondrial biology. So why? Mitochondria are maternally inherited, strictly maternally inherited. They contain receptor for sex hormones. They contain the limiting step for most sex hormone synthesis. So there is no way there is no interaction between mitochondria and sex. And the sad part of it, most of what we know, you could safely assume 99% come from studies in mice. But when it comes to metabolism and mitochondria, mice are not human. And one of my favorite meta-analysis review article by Dr. Pickard's group, they reviewed actually all published mitochondrial data that looks at sex and mitochondria. And out of 1,200 papers, there were only 50 papers that specifically and addressed that question in human. And the results we don't have time to go over are more variable in human rather than this dogmatic approach in mice where female do X while male do B, and so on and so forth. A quick example there, if you just do unsupervised integration, you throw in your RNA data, your metabolomic data, and you see how you get modules that contain gene and metabolite that vary together, and you can correlate them with whatever laboratory or clinical feature you want. And the strongest correlation of all these modules seen was between the pink module, which is a random color, and sex. Then you can look to see what's in that module. And that module actually contains a lot of long non-coding RNA that we're just starting to understand, and along with other genes as well. So to summarize, mitochondria are multifaceted organelles. They serve diverse functions, diverse behaviors. There is no mitochondrial binary approach to mitochondrial dysfunction. There's likely mitochondrial involvement in any muscle disease, let alone myositis. And most importantly, mitochondria play a major role in triggering or defending against immune system activation. Although that sounds complex, it also offers a lot of opportunities for therapeutic interventions, with the main goal is actually to boost mitochondrial fitness, no matter from which side you're going to approach that. And to achieve that, there is a critical need for us to work all together, physician, basic scientists, translational scientists, patients, because there is a huge disconnect between the basic science and the clinical field, as you all may know, in order to move the field forward and make an impact on patients' quality of life. Lastly, I would like to thank all my mentors, collaborators, everyone in their lab, and all the funders as well. And thank you for your attention.
Thank you very much for this marvelous talk. We have time for one or two brief questions.
Please come to the microphone.
Thank you very much for your very nice and interesting presentation, Fabrice Espinosa from Sweden, Karolinska. My question is regarding the reactive oxygen species. Is it is known that they can appear, or everything can be dysregulated before the inflammatory infiltrates can appear in the muscle tissue. Could you comment on that in patients with dermatomyositis and or polymyositis?
Sorry, there was some echo. Do you mind just repeating?
Yes, if the reactive oxygen species can appear before the inflammatory infiltrates in patients with polymyositis or dermatomyositis, because that's known in animal models, but is it known in patients with myositis?
Yeah, I don't think we've done studies or we have the right model to say if they could appear before. Could they appear before? Yes. I think the interesting thing there now
Are we
that
testing this
reactive
microphone?
oxygen species are
Yeah,
more
it's working.
perceived like negatively, they're just
Fantastic.
damaging. But now, as we know more and more, they're actually very important signaling molecules, and they could appear before, I guess,
Okay.
to
We are
actually
on time, so we
induce
stop now
some
because we
stress
have to move on.
response that's healthy to the
Welcome,
cell and
everybody,
help the
to
cell
this
build its resilience.
patient-led session.
So in summary, ROS
First
are
of
not
all, welcome
necessarily
to
the bad
Gcom
players, even
for
in
all
the
the
science of aging.
patients,
Actually,
all the
when you measure it in a
caregivers and
human, it
the
doesn't
professional
turn out to be a
healthcare
huge issue
professionals
there.
and
They're
everybody
more
who's
helping you
joining us
build
today.
resilience and
You have
teaching
probably
your
seen in the
mitochondria and
program
your
that we
cell
are
how
going
to
to have
better handle stress. They are very important signaling
5
molecule
different
to trigger those
patient-led,
original defense
sessions.
responses.
And move on.
Thank you. We have
And,
time for a very brief
start today
question and
with this.
answer. No? We have one? Okay.
With this
Thank you very much for the great
session
talk.
about
I'm wondering that
holistic approach
your main discussion today was
for
focusing on
myositis patients.
muscle myocyte.
So,
right? Is there any
we'll
evidence that
see.
there is mitochondrial dysfunctions in
We are
immune
going
cells, such
to
as T cell, B cell, myeloid cells?
take a little
That's a great
mental
question.
metaphor.
And when we
I
do
want to
bulk
know about
RNA-seq,
your
we just
expectations
automatically think
for
muscle,
this,
but actually it's all the cells in that
session
tissue.
and what
And
we
you're
are
right.
curious to
Actually,
know
to
what
learn
you are
more, we
going
have
to
to look
expect
at
from this,
individual cells, especially in
Gcom
myositis,
this
where
year.
the cell play a very
My name
important
is Anneli
role.
Gcom,
For
and
instance, in
I'm
IBM,
from
actually
Sweden.
determining the
And with
CD8
me, I
fate
have
is actually that
Paula,
switch in
and she will
metabolism
go up here.
that dictated by mitochondria.
Thank you,
When they
Anneli.
activate, they rely on
And
glycolysis,
don't go far.
and then chronically
I
they're
join
more
Anneli in welcoming you all to
fatty
Gcom
acid
and to the
oxidation.
first
So
of
you're
our
right, it's
sessions
not
and
just
workshops in the
muscle, it's
patient-led
also the
track,
immune
open to
cell
all,
and the
but
interaction
specially
between
designed to
muscle
help
and immune
those
cell,
of us
and not
navigating
that just, and
myositis
the
journeys
extracellular matrix
flourish
and
in
so
those
on
journeys.
and so forth.
So we
that
are,
result
many of us are
into
here
what,
physically
the
in
disease look
Lisbon,
like, or the
but we
pathogenesis
also
of the
have a number of
disease.
colleagues
thank
who
you
are
for
connected
pointing that out.
remotely via the virtual link. And
Okay.
so our
Thank you
colleague
very
Linda
much. I'd like
Kober
you
is
to join me
monitoring
in
that,
thanking the
and
speaker.
we invite our virtual colleagues to complete our introductions as well. So we're interested in
And we're
knowing
gonna
from
move
our
now
online
to Dr.
attendees
Katarina
name,
Almeida.
country,
Katarina is an
patient
assistant
advocacy
professor at
organization,
University
if
of Aveiro
applicable,
here
and a
in Portugal,
myositis diagnosis.
specializing
And
in
for
the
everyone
innate
in
immune
person and
system
online,
and how
please
cellular
join
homeostasis
us in
shapes
our poll
immune
question.
function,
You
its
can
role
scan.
in tissue
Look at
repair.
that, Anneli. They all got it. Good. Phones up.
hello.
Cameras scanning.
Good afternoon, everyone.
Tell us what you are hoping
Do I
to
need
get
to do
out of
something for
Gcom
the
2026.
presentation?
And we should
here we
see
go.
the
Okay,
results.
so good afternoon, everyone. It is
Refresh.
truly a privilege to be here. So I must first of all thank the organizers for the invitation
Slowly, as
to be
folks
here
come
in this
in.
amazing meeting. And today I'm going to tell you a little bit on the
And,
role of
I
interferon
know
dysregulation.
one thing that I'm hoping to get
And
out of
this is one
Gcom,
of the
Anneli,
first
is a little
talks of the
more
conference
understanding
in a
of
session dedicated to
artificial
pathophysiology.
intelligence at
So I'm
one
going
of
to divide
the
the talk
patient-led
into
workshops
parts. In the
tomorrow. We
first
have
part, I'm going to tell you a little
experts
bit
sharing
of what
about
is
artificial
interferon, what
intelligence
triggers
in myositis, which I
interferon
think will be very
production,
interesting.
and what are the consequences when we have pathogenic levels of this interferon. And then in the second part of the talk,
I am
I'm
trying
going
to
tell you all a
refresh the
little bit
Minty
of the
poll,
results,
so
on
keep
the results that
putting
we've been getting in
your
the lab
responses in
on
there.
what can regulate type 1 interferon production.
You guys are going to like it when it refreshes.
And,
I have a
must
sneak
start by
preview
disclosing
here.
that I have no financial conflicts of interest, but I also want to Disclose that I'm a biologist
Look at
trained in
that.
immunology,
Ooh, and it's
so
moving
I'm
around.
not a medical doctor.
I just love it
and
when
thus this
technology
seminar
works.
is mostly
Special
gonna
thanks to
focus
all the
on
really
biology
gifted
of
technology
interferon.
experts in the back of the room.
As you know
and
better than me,
with us, thanks to
myositis
EEM
is
as
highly
well, the
heterogeneous.
meeting
You have
organizer
many
for
organs that
iMyos
can be
and Gcom.
affected. And
Okay,
the
it looks like
intensity
most
that these organs
people
are affected
have
is gonna
updated
vary
or
for
entered their
different
responses.
subtypes of
So
disease and also
we see a
for,
lot of
different
alignment
patients, even
around
within the same subtype.
hoping for knowledge,
So it's
networking,
very
and
important. It's hugely
connection
important
here
to
at
be able to
Gcom.
stratify
I
the
think that's probably a
patients, to
pretty,
find ways to
pretty
monitor
representative
treatment,
response,
and
Amelie.
hopefully to
So,
personalize
thank
this
you all,
treatment.
and welcome
And the
everyone.
interferon
Now,
here
just so
will
we
be
can get to know the
important
in-person
as it
audience a
provides
little bit
molecular
better, we don't have
access
time for every
from
single
innate
person to
activation
introduce
to
themselves,
pathology.
sadly.
So
Find
it's
me at the
important
welcome
to
reception
understand
later,
a
though,
little
because
better
I'd
how
love to
this
know everything
interferon
about
works,
each one
as
of
this
you.
can help
In the
us
meantime,
finding
let's just do maybe a
biomarkers
quick show of
and,
hands.
maybe
Any
even
other
therapeutic
Swedes here,
targets.
Annalie?
So what is
Oh,
interferon?
okay, yeah, yeah.
Interferons are
I know I have some
cytokines that can have a
Americans
role
here
in
as well.
antiviral defense,
Yes, show of hands.
but
Okay,
also,
great, great.
in
Let's see, I see
activating the
a
adaptive immune
Swiss friend.
system and
Yes.
triggering inflammatory
Oh,
responses.
who else?
And
Oh,
you can see
Turkey?
here in this figure that there are
Yes,
three main
okay.
classes of interferon. We can have type 1,
Kind
type 2, and
of,
type 3.
yeah. I would also
And on
like
the
to be
left-hand
kind of
side,
from
we have
Turkey. What a
type 1
beautiful country.
interferon, which
Okay,
includes
any other countries
interferon alpha
represented? Would
and
anyone like
beta,
to share anything
which
quickly
will
aloud?
bind
Yes.
to the
Oh, yes.
interferon,
The Netherlands.
IFNAR receptor.
The Netherlands.
And Netroderma,
Marie Christine.
constituted
Czech
by
Republic.
the IFNAR1 and
Belgium.
IFNAR2 subunits, which are associated with JAK1.
Portugal.
and
Well,
upon
our
binding
beautiful
to the
host
ligands,
country.
will then trigger the
Thank you for your
STAT
gracious
signaling
hospitality.
cascade, which will lead
Okay,
to the
terrific.
expression of different
We will move on
interferon-stimulated
into
genes or
the educational
IGs.
content of our workshop,
And then,
but we
so
have
this
another
type 1
super
interferon, I
fun
must
polling
stress, is
question
usually
at the
associated
end,
with an
so
antiviral
don't worry, there is
response.
going to be a
In the
lot
middle,
of
we have type 2
interactivity
interferons.
in today's
And here we're
workshop
talking about
about living
interferon gamma,
holistically with
which is mostly
myositis,
produced
balancing
by
conventional
T
drug
cells and NK
therapies with
cells,
functional
and
medicine
will be
and lifestyle
involved in
changes.
macrophage
And I know that we're all here
activation
in
and
Auditorium 8
inflammatory
and
responses. So
online
now,
because
this
we know that the
interferon,
value of
will
patients
bind to the
attending and
interferon gamma
contributing to the
receptor,
program in Gcom
which is
has been
again
recognized
associated
ever
with
since its
JAK1,
inception
but also
in
JAK2,
2015.
triggering STAT
With subsequent
signaling
Gcoms over the
cascades,
year, there
and
has
again
been
leading to the
increasing
production
interest
of
and
ISTs, but
participation
these will be
by
different
patients,
ISTs
care
from the
partners,
previous
and the
ones.
organizations like
And then on the
TMA that
left, we
represent
have
them.
type 3
And so
interferon.
here at Gcom
These are
2026, patient
interferons that
voices will be
include the
echoed
lambda
throughout the
family,
daily
and they
program
will have
with
a
patient
very similar
perspectives shared
function
in
to
the
type
scientific
1
sessions.
interferon, apparently.
And here,
the signaling cascade,
patient-led
you can see, is
workshops
very
and
similar,
sessions, as
again, to the
Annalie
type 1
alluded to. In
interference,
total,
but the
we
major
are offering
difference
more
is
than
that the receptor
550
for
minutes
type
of
3
dedicated
interferon
airtime
is more
from
localized.
patients
This
and for
receptor
patients,
will
and
be
this emphasis
in various
ensures
sites.
that
And
all
today,
participants,
for this talk, for the
650
sake of
plus,
time,
here in
I'll
Lisbon,
focus
plus those
mostly
online,
on what's known
will
regarding
benefit
type
from
1
the vital
interferon.
perspectives of the individuals
So
with
what
lived
drives
experience
the
in the
production
forms of
type 1
myositis.
interferon? As I mentioned,
So
this,
again,
the
welcome and
evolutionary
welcome to
role
our
is
experts
mostly
and the clinical
antiviral
support
defense.
faculty for this
So
session
the
in
production
particular.
of type 1 interfering
Annalie and
interferon will
I
be
have no
triggered
disclosures.
when,
We do have
pathogen
some
recognition
housekeeping
receptors
requests. This is
will
not one
bind
of the
to
really
their
fun
ligands in the
interactive elements I
endosomes, and
promised
here we
you,
can have, for
but
example,
as a reminder,
TLR7 and 8
silence
and
your phone.
TLR9, which
You
recognize
definitely
the
are welcome to take
degradation
pictures.
products of
In fact, we'll be
single-stranded
taking
RNA
hopefully
or
a group
CPG,
photo afterwards and a
CPG
photo
islands
during our
on DNA.
demonstration.
These two,
Keep chatting
or actually
online,
these
and
three
Linda will keep
receptors
relaying
will
that to
signal
us.
through mediatate. And then in the
And
endosomes,
here
but in
is
different
our
cells, we
agenda
can have
for the next
TLR3,
hour and a half
which
or
will
so
recognize
with the times
double-stranded
breakdown. So
RNA,
it's going to
and will
be
trigger
an
signaling through
action-packed
TRIF. And in the
90
cytosol,
minutes.
we have different
And
receptors.
we can leave the slide up just
And we
another
have
couple of
the one
seconds.
that you
Folks are
probably
taking pictures
recognize
of it.
better, NDA5,
That's good.
as well as VI,
And
which
now
will recognize
Annalie will
double-stranded RNA,
conclude the
and will
introductions,
signal
and
through MAVs in the
then we'll introduce
mitochondria.
our first speaker.
And we
Annalie,
still have
comments?
CGAS, which recognizes double-stranded DNA and will signal
You
through
might
STING.
notice that I have
Independently
also
of what
given you
triggers
all
the activation
little
of these,
paper that you have
independently
all
of the
the
receptors
text
we're
for
talking
this-
about, we're going to
these
converge
slides
to
because we
pathways
think we
that lead to
are going to
activation
get
of
them
transcription
little
factors
bit
such
harder
as
here.
REF3 IRF3
So we have to
and
move on very
IRF7
fast.
that will lead to the
We will-
transcription
I have
of the
this,
type 1 interferon that we're
This
talking
little
about
quoting
here.
from our
And type 1 interferon is mostly
former
produced
patient
by
who is now cured,
plasmacytoid
Lars
dendritic
Nordell
cells, or
in Sweden,
PDCs. These
who-
are the
who is a
professional
psychotherapist,
producers of type 1 interferon, as they
and,
can very quickly
giving
produce large
really
amounts
nice
of type
words
1
about
interferon. But it's
how-
known that other
how do
cells,
you-
including
how do
tissue-resident
you
cells, can also
move
produce this type of
holistic about,
cytokines.
about the thinking.
But I just talked
And
with you about
this
the
picture
infectious
we
triggers
have
of these pathways.
also.
But in autoimmunity, we can also have sterile or
who we
endogenous
kindly got
sources
from,
of
Leslie
immunostimulation,
and Sakatu
such
from
as
the
the immune
Myra.
complexes formed by
You can,
nucleic
study the
acids and
picture because
autoantibodies,
it's so
which will
many
be
things in it,
internalized
so
through the
we don't
endosomes
have
and can initiate this
really
cascade that I
time to
mentioned.
talk about it.
Or we can have immune complexes formed again by the
Thoroughly,
nucleic acids,
but you can
that
study
will
it by
form
yourself.
complexes with alarmins, which are released
now
when we have
we
NET
are going
formation
back
or
to
inflammatory cell death.
Paula
And
and
these
our
can be associated with defects, for
expert
example,
who
nucleic
is
acid
going to
clearance, or
give us a
on
lecture,
the way of
please,
expression
Paula.
of retroelements.
Thank
which
you,
are
Annalie.
viral-like
Excellent
elements that
context
exist in
on
the
what is meant
DNA.
by the holistic
Moreover,
approach.
when
And we
have
really do
tissue
want to
injury,
include
we
every
can have
aspect
release
of being a human
self-DNA or
and being
mitochondrial
affected by
DNA, as was
myositis in
mentioned
today's
earlier,
session.
that again
All of
can
our
engage
presenters
these
are
pathways, in
open to
particular the
questions,
CGAS
so
STING
submit
pathway.
questions at
So
any
when these
time, and
pathways
presenters will
are,
answer them at the conclusion
engaged,
of
as
their
I mentioned,
presentation.
we're going to have
So for
triggering
the
of
first
signaling
part of our
cascades leading
workshop,
to the production
we will
of
have
type
an
1
overview
interferon,
of functional
then
and
we have
lifestyle
an
medicine
ISG
from
program,
Dr.
or
Sherb
interferon
Bilichalar.
stimulated gene program.
Dr. B is
So we can
professor of
measure
neurology
type
and
1
co-director
interferon in
of the
different ways
neurology
in the
residency
patients.
program at
We can
McGovern
measure
Medical
directly the
School,
protein
University
in
of
the
Texas
serum
Health.
or
She
the
is
plasma
board
of these
certified in
patients. We can
neurology and
measure the
neuromuscular
levels
medicine,
of-
and she
we can quantify
specializes
the
in
levels
nerve
of
and muscle
interferon alpha or
pathology.
interferon
Her
beta.
clinical and
But
research
these,
interests
the
include
amount
muscular
of these
dystrophies and
cytokines is usually
PIIMs
very
and
low. It
advancing
can be
care
very
for
transient, and
neuromuscular
the data
disorders.
can be
We are very pleased to
difficult
welcome Shore,
interpret.
and we
So
know we
other
will enjoy
methods have
learning
been
about
developed,
lifestyle
and
medicine
many
and
labs
its 6
actually
pillars.
present what's called
Please.
an interference signature, which is a score that is calculated upon measuring by real-time
Thank you for having
PCR
me.
the
It's a
expression
pleasure
of the
and a
interference
privilege
stimulated genes.
to be on
This
this
is a more
podium.
robust readout, but there's
So
currently no
I practice
standardized
at,
method in the community.
in Houston,
And it
Texas, in
matters
the
to say that
U.S.
we can also look at the
So our
tissue
healthcare
levels
system
of
is a little bit
interferon. And a common
different
way to do this is to look
from
again
a
to
lot
the
of the other
expression of the
European
ISGs
countries
in
or
these
little
tissues. And you can
European
find
countries.
high expression
We
levels
have
of some
mostly
ISGs
private
in the
insurance,
muscle
and then
of patients with
for
dermatomyositis. So
our
what
patients
distinguishes the
who are
protective
over
from the
65, we
pathogenic
have
levels of
Medicare,
type
and
1
then for
interferon?
patients who are
As I
younger
mentioned
than
already a few times, we need this
65
type 1
and who cannot
interferon
afford, we have
for our
Medicaid, but
antiviral risk
unfortunately,
defenses. And
our
indeed, we have an
healthcare
acute
system
when we
needs
have
a
a
lot
viral
of
infection or some
improvement.
intracellular bacteria as well.
And our
we'll
patients
have an
and
acute response where we
us as
increase the levels of type 1
physicians and other
interferon, we increase
healthcare
sensitivity, but upon
providers,
reaching its
we're
peak,
sort of stuck.
the levels
And
will decrease to the
what
basal
happens
levels because we stopped having the
during
trigger,
our
or
interaction with our
or
patients,
we
we're
have
almost in a
resolution
rush.
mechanisms for this response.
We have
But if this fails,
maybe
if
10, 15
resolution
minutes or
fails, and
sometimes
if
20
the trigger
minutes or
continues
so
to be present,
to go over
we can have a
everything
persistent
and to be able
response.
to
So the levels of
come up
interferon will not
with
decrease,
useful
they'll remain
recommendations
high,
for our
and this is when we
patients,
start having chronic
and
inflammation
also
and tissue damage.
advocate for them.
So I said
And
here that the levels
in the
can
end,
decrease when we have an acute response, but this is not
there's a lot of
passive
frustration
mechanism.
because
This occurs because we have negative
we're able to talk
regulators
about
in our system
medical
that
treatments,
will contribute
but we're not able to really approach our
block the interferon
patients
response and to
from
return to
every
basal
other
levels.
angle, such as
So in
nutrition,
this figure, you can see
exercise,
the scheme of
because
the
we know that
cascades
those
that I
are really
mentioned earlier.
important
And in red,
in
you have some of the
making
inhibitors
sure
of
we're
these
optimizing
pathways. So we have
our patients'
inhibitors
well-being
acting
and
on
also
the innate
response to
sensing
treatment.
cascade and also
Therefore,
on the JAK signaling cascade.
American
And
Academy
usually we have these
actually now they
breaks
have
acting to
an American
control the
Academy
levels
of
type
Lifestyle
1
Medicine.
interferon.
And
But as you know,
you
type 1
can
interferon
be a
levels
physician,
can be
you
high
can
in
be
different
a
autoimmune
physiotherapist,
conditions. They can be
you
high
can
in
be
lupus,
a social
Sjogren's,
worker. There are
systemic
different
sclerosis, and
tracks that you
dermatomyositis.
can
And
actually
although
take, and you
talking
can be
about
board
type 1
certified in
interferon, all of
American
them, the
Academy
tissues that are affected seem
of
different.
Lifestyle
So you
Medicine.
can have high
And
systemic levels in
we are
lupus.
also in the process
In
of getting
Sjogren's,
grants
you can have high levels
from the
of type 1
Lifestyle
interferon in
Medicine
salivary glands.
group,
In
so
systemic
that
sclerosis,
we
the
can be
levels of
funded
type 1 interferon have been correlated
and we can spend
very early, from the
enough
very early,
time with our
signs
patients.
of disease
So what
to
is
fibrosis
lifestyle
development
medicine?
later on. And in
So
dermatomyositis, we have
lifestyle
increased
medicine actually
type 1
has
interferon
6
in both the
pillars.
skin and the muscle.
And let's talk about those pillars, and let's
indeed,
talk about how
different
we can
studies have
translate
looked at the
them
levels of
into
type 1 interferon in
myositis-specific actions.
dermatomyositis, and
So
it
these
has
are
been known
the
for
6 pillars.
some time that
And,
these scores are higher in these patients.
So
And this
nutrition
is just one
is one of
example
them,
of the
and
studies that
Dr.
have been
Gupta is
performed. In this case, the authors
us,
looked at muscle
very
biopsies,
detailed,
and they performed
lecture about
RNA
that. Physical
sequencing in these
activity,
muscle biopsies.
definitely.
And as you can
Dr.
see
Sir is
here,
going to
when they
talk
looked
about
at expression
also.
of different ISGs,
Stress, managing
they found
stress is
that
another
the
pillar.
dermatomyositis
Relationships
was the
is
subtype
another
of
connection. Sleep
disease with the highest
and
levels
avoiding
of type 1 interferon. However,
dangerous
they also
substances
looked
such
at
as
type 2
smoking,
interferon in these genes,
drugs, and
here the
alcohol.
pattern is a little bit different. And this is to call your attention that
I'll
type 1
briefly talk
interferon is
about
mostly
nutrition.
associated with dermatomyositis.
American Academy of
but
Lifestyle
the other
Medicine
subtypes of diseases
recommends
will
a
be
whole
mostly
food,
associated
plant-forward
with other types
pattern,
of
nutrition,
interferon that I'm not focusing on today.
like based on vegetable fruits, legumes, whole
So
grains.
the literature on dermatomyositis is still a little
American
bit
Academy of
sparse,
Lifestyle Medicine
but we
again
can try to
suggests
build a
that
model
we
on what might
maintain
be
our
going
protein
on. There's
intake
still many
because
gaps,
protein
as you know.
intake is also
But still
important
we can
when we
think that,
do our
we know, for example,
exercises,
that
physical
dermatomyositis
therapy
does indeed
to
have
maintain our
high
muscle
levels
mass.
of type 1 interferon.
And there is also
possible
this
routes
notion
for the
that we can
sensing,
get that protein
maybe
from
the TLR7,
meat,
TLR9,
which is not
MediA5,
accurate.
this is one where we have
And
most
that's what
evidence, I
American
think.
Academy of
And
Lifestyle
then
Medicine is
C-Sting
suggesting.
is emerging
But when I
as a very interesting
counsel my
route,
patients, I never
and
tell them,
contributed
you have to be a
the
vegetarian
development, of these
tomorrow,
high
and that's the only
expression
way
levels of
forward.
type
Of
1
course not.
interferon.
Everything is
And
on
it matters to
moderation.
say again
But
that
whatever
PDCs
you can
are the
do
cells where we have the
to
strongest evidence as being
improve your
relevant
health.
for the production
And,
due to their
and of
capacity to
course,
produce high
limiting
levels and
ultra-processed
very high
foods,
levels
which
of
is
type 1 interferon, while other cells may
probably
also
like
have a role.
tougher for us in the
So
US.
as I said
You
earlier,
guys are
when
better
we have these cytokines,
here.
will trigger the
And
JAK-STAT signaling pathways,
of course,
which will lead
when
to
nutrition,
these interferon
when we
score
talk about
signature,
nutrition, we
and
also
we're gonna
talk
have
about dysphagia
persistent
and what
high
we're
levels of
supposed to
this,
do, refer
signature.
to speech therapy,
So what are the consequences
maybe
of
texture
having too much interferon
modification.
in our body?
And
Well, if we have
of
too
course,
much
an
interferon in the
excellent
tissue, this can
lifestyle
contribute
medicine
to immune
clinic
cell
is
recruitment
going
and
to
immune
be
cell activation.
a
We're gonna have
multidisciplinary clinic.
certain
So
chemokines
your
being
neuromuscular
highly
specialist is going to be there
expressed,
in
which
one
will
room.
recruit other cells.
The
In
next
this
room
case,
is
we're
going
gonna
to be
have
your speech
the
therapist.
CXCL10,
In the next room, your physical
CXCR3
therapist, your
axis
nutritionist,
involved.
social
We're gonna have
worker,
increased levels of
and
MHC
your
class 1
pulmonologist.
and
So all of them
higher
will be
antigen presentation, which
in
will
the same
lead to more
floor or in the same
higher,
clinic.
immune
But if not,
activation and
at
activation
least
of adaptive
if you
immunity.
have
But these cytokines
good
can also affect the tissues. They will
interactions or
affect
connections with
endothelium.
all these specialists,
Type 1
it's
interferon
definitely
has
going
been
to make a
found
big
to
difference
impair
in your patients
angiogenesis
or in the
and impair
patient's care.
capillary formation.
And that's what
they
we're
will
trying
also, or it will also affect the
do in the US and
myofibers,
especially
since
at
type 1
UT
interferon
Houston,
blocks
Texas.
myotube
So pillar 2 is
formation,
exercise.
And it has been found, at least in in vitro
the evidence,
systems, to be
and we have
associated,
experts here, Dr.
with
Anderson is here,
atrophy
so
programs,
structured
in
endurance and
myofibroblast.
resistance training
So
improves
for the first part
strength,
of my talk, the take-home
endurance
messages
and
are
quality
that
of
type 1
life in
interferon is
myositis
essential
patients.
for antiviral
Unfortunately,
defense,
still
but
in the US, there's
and
this
it also contributes to anti-tumor
notion
immunity.
that
But when
exercise
persistent,
is
this
dangerous.
type 1 interferon will
And
become
I get a lot
pathogenic
of
and has an
patients, they
important
come to
role
me, they're like,
play
oh, Dr.
in
B,
different systemic autoimmune
my
diseases.
physician or
So this
therapist told me to
interferon program
stop
can drive
exercising
immune
because
activation and tissue
all
dysfunction.
inflammatory
So it's very important to
myopathy, which
understand
is
what
now we know
triggers
that
the
that is
sensing
not
that will
accurate
lead to
information.
type 1 interferon production,
We need
what
to
triggers
guide
its
our
signaling,
patients,
and also what are the
and we need
molecules
to
involved in the
customize
negative
their
regulation
exercise
of
program.
these pathways.
And
this knowledge has already
again,
led to the
as
generation
physicians,
of several
we have to start the
potential therapies
process,
that you
and we
know
have to
better
collaborate
than
with
me and
our
are here depicted in this picture.
physiotherapists
But the key challenge
plan,
still remains to know exactly
the
when,
perfect
where,
exercise
and
schedule
how
for our
much
patients.
to intervene without
And
compromising
of course,
host
initially,
defense.
this needs to be
So in our
supervised
lab,
so we
are working
can
on a little
route
bit
for
more
our
also
patients,
on
we
the
can cheer for
mechanisms
them,
that regulate
because
the
one of
production
the
of type
main
1
reasons
interferon.
why
And we're doing this
the
by
patients
focusing
cannot
on
sustain,
plasmacytoid
yes, there are
dendritic
several
cells that I
barriers, right?
mentioned are the professional
Getting there,
producers
getting
of
maybe
type 1 interferon.
you need to go
so
to your
despite being only
therapist
a
at
very
least a
small
couple of
percentage
sessions.
of cells antivirals in our blood.
Sometimes
They are
affordability,
crucial
there are
for
several
antiviral
things that can
responses, and
factor
they have an
into.
important
But one
role
of the
to
big
play
reasons
in
why
autoimmunity.
our patients cannot sustain
And this tells
exercise is
because they
because
produce so much protein so
they
quickly.
start
They
and
possess
then they
a
lose
very
the
large
habit.
endoplasmic
So initially, if you
reticulum, or
supervise
ER.
them,
But
the
it's also
adherence
known
is
that they
going
have
to be
basal
better.
activation of ER responses.
And then that's also what
So
we're
what are
working
these
on.
ER, so
We're
ER
working on
stress
a
responses. What are these ER stress responses? These are the responses
app.
that the cell
Again,
initiates
we work with
when
Dr.
we
Anderson
have
again
accumulation
and
of unfolded or
UT
misfolded
Houston, Rice
proteins.
University in Houston,
So in order to restore
and
the
Karolinska
intracellular
Institute.
balance,
The app is
cell
ready,
will
but there are
initiate
some
the
bureaucratic
unfolded protein
obstacles
response,
we're
which
trying
is
to overcome.
schematically here. This unfolded
In our
protein
institution, there's
response,
some
or
legal
UPR,
jargon that we
has
have
three
to
branches.
come out with.
The PERK branch,
But the app is ready, and then the
branch,
idea
and
is
ATF6.
we're going to, with
When
this
we
app,
initiate
we're going
these
to recommend
branches, we're gonna
the
end up
exercises,
by
and we can see
blocking
on the
protein
dashboard
synthesis,
what our patients are
by
doing, and we can cheer for
stimulating
them,
degradation
and we can
of
basically
mRNA, and also producing different chaperones.
monitor them and also
altogether,
help them
these
through
will
their
contribute to
exercise
restore
journey.
the balance within the cell.
So I'm gonna,
And we wanted to know whether
I think
these
Fulden, are you going to talk about
UPR or ER stress
exercise, right?
responses could impact the activation of the PDCs. And this work was pursued by two brilliant people in the lab. One of them is here in the audience, Beatriz Ferreira. She did her PhD with us, but she's now taken a new challenge in myositis. And they started, both Beatriz and Daniela started by treating the PDCs with sub-AB, which is a toxin that initiates ER stress. So they isolated PDCs here from healthy donors, treated them with sub-AB to induce ER stress, and found that there was an increase in the production of both interferon alpha and interferon beta. But this only occurred in the PDCs and not in the negative fraction from the isolation procedure. So we wanted to study the molecular mechanism that was behind this effect, but as I mentioned earlier, PDCs are very rare and it's difficult to work with them. So we started working with a PDC cell line, which are called Cal1 cells, that are easier for us to manipulate. So we treated this PDC cell line with sub-AB, and first we tested whether the ER stress pathways were being triggered, and they were. So we could see phosphorylation of phospho-ELF2 as well as blockage in protein synthesis, and I'll come back to this later, which showed that the PERK pathway was initiated. They also found XBP1 splicing increasing, pointing to the IRE1 alpha pathway, and BIP expression increasing, pointing to the ATF6 pathway. And again, when they treated the Cal1 cells with this sub-AB, they saw an increase in interferon beta. And interestingly, if the cells were treated with a PERK inhibitor, so if we inhibited one of those branches from the UPR, we saw a reduction in this effect. Also, when the cells were treated with ISRIB, which blocked the effect on protein translation, we no longer saw this effect on type 1 interferon. Moreover, we wanted to know if the innate sensing receptors that I mentioned earlier could be involved in this mechanism. So we performed experiments with a MAFS knockout cell line, but the cells responded the same way. However, when we looked at the STING knockout cell line, we saw that the effect was not as strong as in the wild type. Also, when using a STING inhibitor, we saw that the effect was very reduced compared to the untreated cells or treated only with sub-AB. So this induction of type 1 interferon induced by ER stress requires both PERK from the UPR pathways and STING, an innate signaling molecule. As I mentioned earlier, we know that we're blocking protein synthesis. So our hypothesis next was that we could be interfering with the levels of the negative regulators. So if we inhibit the expression of the inhibitors, we can potentiate the signaling. So Beatriz and Daniela looked at the expression of several of the known negative regulators, and we found that one in particular had the strongest result. In fact, TAX1BP1, which is one of these negative regulators, showed a strong reduction on its levels when the cells were treated with sub-AB. And interestingly, if the cells were treated with ISRIB, a condition where we no longer saw the type 1 interferon being produced, we had higher levels of tax1 BP1. So our current model is that by inducing ER stress, we are interfering with protein synthesis, thus inhibiting the levels of the protein of the negative regulators, in this case tax1 BP1, which allows us to unleash the innate signaling and thus have production of type 1 interferon. So in a parallel project that we have been running in the lab, we had Fatima looking at a slightly different side of the equation. Fatima pre-stimulated the cells with an ER stress inducer and then stimulated the CGAS STING pathway. And again, we saw an increase in the production of type 1 interferon. We're still exploring the mechanism behind this effect, and here we're using a slightly different approach. Now we're exploring the interactome of STING. So Fatima built a STING turbo ID fusion protein, which allows us to know which molecules will be in the vicinity of STING when the cells are treated either with CGAMP to trigger the CGAS STING pathway or with the ER stress inducer. And indeed, we found many differences. We found many proteins that were altered, but we're still exploring this, so I don't have more details to share with you today on this part of the work. But what is important from this work is that induction of the UPR, in particular by sub-AB, will drive type 1 interferon expression in a PERK and STING-dependent manner. But we still wanted to know what is the impact of this mechanism in the tissue. So our next question was whether these PDCs could affect fibrosis. And Beatriz did something else. She prepared co-cultures of PDCs with fibroblasts. Here we used lung fibroblasts. And she saw that the cells establish really strong intracellular conjugates that are not easily separated. And then she treated the cells again with sub-AB. And making a long story short, what she found is that when we induce ER stress in the co-cultures, there was an increase in the production of both mRNA and protein levels of fibronectin and alpha-SMA. We have more details of this story in a poster. I don't have time to present to you here today. So if you want to hear more about this effect of ER stress-activated PDCs on fibroblast activation, I'll be in a poster session on Wednesday. So from the talk, the take-home message for today are that type 1 interferon is very important for antiviral sensing and antiviral responses, but it will become pathogenic if it's persistent. The triggers for this kind of response are not only infectious, they can be sterile, and we saw some of these triggers. And in dermatomyositis, there's a domination of type 1 interferon response, which can affect tissue damage. Pathology in the end will reflect this balance between the triggers that we have, how long they persist, and whether the negative regulators are acting or not. And from our own data, I hope I convince you that ER stress can amplify the interferon signaling and reshape the negative regulator landscape, and that stress-activated PDCs may actually also promote fibroblast activation and potentially fibrosis. So the most important slide in the end, the acknowledgements. So I'd like to thank everyone in the lab who's been involved in this work, but particularly to Beatriz, Danielle, and Fatima. Philip is a long-time collaborator, also important for us for the work we do in the lab. And Mariana Machado is the person who's taking over this project and exploring the details in further detail. And last but not least, the medical team with whom I'm collaborating. You may have noticed, and you may ask me if I tested these in patients. Well, we're working on it, and I must acknowledge these people from the hospital in Aveiro who have been essential in setting up these experiments. Dr. Anabela Barcelos, Dr. Carolina Mazeda, Dr. Eduardo Dourado, and the nurse Catarina Silva. So thank you all for listening, and I'm open to questions.
6 pillars, and basically when
Thank you
when
very
our
much
patients
for
come
this
in, we
beautiful
have
talk.
to
Please come to the microphone
talk
if
about
you'd
these
like
6
to ask a
pillars with
question. We have
our
time for a
patients,
few
not
quick
just,
questions.
okay,
Short
so how are you
questions,
doing
please.
in your myositis?
Thank
Okay,
you very much for the wonderful
this
talk.
is the medication,
So
take
you
your
mentioned that the
prednisone,
lupus
IVIG,
and
this and that,
dermatomyositis
increase the
is
dose,
both
and then
interferon activated
let me check your
diseases.
strength, and then you're
But
good to go.
the
No, we have
pathogenesis
to talk
and
about all
the
these 6
symptoms are very
pillars because
different.
those are
right?
also
So do you think
important
any differences in terms of
our patients'
interferon in both
prognosis
diseases?
and how
It might
they
be a bit tricky
respond
question.
to our
It is a
treatment.
tricky
And also,
question, that one.
it helps with
So yeah, definitely there are
adherence of
differences
our
in the different
recommendations.
subtypes.
And
You're gonna have
our
different
patients also need
signatures,
to
so
ask
it's
these
very
to
likely
their
that the
doctors, and they
pathogenesis
need
will
to
be
request
different.
and
I
actually
think
demand
we have
that we
clues for the different subtypes
provide
of what's
care
going on,
like
but we
this.
still don't have a
We
clear
have to
picture.
change
And
how we
for me, it's
provide
really
care.
interesting
And of
that
course,
we
it
have
all
such a-
also, when there's
so
demand,
these
the
subtypes of
system
diseases have
has to
such
change.
a different behavior. And
Other
trying to
special
understand what's
issues in
happening.
myositis
But I don't know the
patients,
answer. I don't know if you guys know the answer.
photosensitivity, of course, always we talk about sun
Maybe I
protection,
can
fall
use
prevention
Chair's
is important,
prerogative
and
to
that
lead on
goes with
from that question and ask
exercise.
you
This is
about
something that I
reduction
always
of interferon.
discuss with
You
my patients.
focused on
What can we do?
PDCs, which of course
And
in
then
the
if
healthy
any
functioning immune
breathing
system
issues, of
are a
course,
key
talk with
source of
our
interferon. But actually in
respiratory
myositis we
therapist
have
and
good
then
evidence
determine
for
what
the
they need.
unfolded protein
So this is
response in
like
muscle fibers as well as
12-week lifestyle
mitochondrial
plan
DNA release and other triggers
that
of
we can
interferon
start,
production.
and then
So
we
have
can
you had a chance to look at these same assays
basically
in myofibers
escalate
from patients or in culture
every
systems?
That's what we're
couple
trying
of
to
weeks.
set up in the moment. We haven't done
And
it
of
yet,
course,
but
if
that's
our
definitely in
if
the
our
plan.
patients
There's an important
they need
point
more
here, which is also
support,
the type of
or
interferon that the
they
cells
use a
produce.
walker,
Interferon
they're in a
alpha
wheelchair.
or interferon
So
beta.
we
So
basically
we still need to
customize
clarify which
our
interferon
program
is really
and then take it from
relevant
there.
and which
So
one
we
is
can come up
produced
with
by
this
each cell type.
lifestyle
Probably
plan,
PDCs will produce
talk about
interferon alpha, and then
the
the tissue
nutrition,
resident
talk
cells
about
will
the
produce
sleep, talk
interferon
about
beta. But we in our lab haven't
exercise,
checked this
all
yet,
of that.
and the impact of the ER stress in the tissue resident cells we haven't done yet, but we're now setting up the assays to do it. I hope we
So
can answer
what
this question
you
soon.
can ask
I
your
totally
physician,
agree. And maybe the type 3 as well, which
so
Yeah, for sure. Yeah,
they can
yeah.
give
So in
you
vitro,
questionnaires.
with these
You
PDCs,
can
we've seen type
ask your
3.
physician,
I didn't show
hey,
it today, but
I
we
want to
see
talk
it.
about my sleep. If you
Thank
don't
you.
have
Any
really
last
enough
quick
time,
question?
a lot of time, why
Otherwise, I hand back to
don't you
my
come out with these
co-chair.
like
Thank you very much
questionnaires?
for
There
a
are sleep
beautiful talk.
questionnaires. We give our patients these sleep questionnaires, and I can fill that in beforehand. I can fill that in my quality of
We
life,
now
my
proceed
depression
with the
scale, all of
presentation
that
on
we
B
give
cell and
to our
autoantibodies
patients.
at the center of pathogenesis.
And then again, shared medical
by,
appointments.
Oliver Bouillet.
The way to move
MD,
forward
PhD,
is
full professor of
base the
immunology
clinics
at
on
Rouen
these 6
University
pillars and also
Hospital
do it in a
France, and
multidisciplinary
director
way.
of the
Multidisciplinary
INSERM.
clinics, we
You
have a lot
went
of
to
evidence, for
3-4,
example,
and
for our
that's
ALS
exactly like
patients.
that, research
There
lab.
is
His work focuses
really
on
good data that a
immunology,
multidisciplinary clinic
biotherapies, particularly in
actually improves
myositis, including
the
translational
longevity
research,
of our
clinical
ALS
trials,
patients
and
and also
innovative
their
therapeutic
quality of
approaches.
life.
Please.
I definitely believe I'm a big
Thank
proponent
you
of
very much, John.
multidisciplinary clinic.
So we'll talk about B-cell
and
in myositis
if
today.
there is
and,
room, of course,
well,
it
here are
depends
my
on
disclosures.
the
Well, being an
logistics,
immunologist, I
funding,
guess
all of
that if I
but,
tell you that B
a
cells are a
multidisciplinary
type of
clinic where we're
lymphocyte
going
that
to
produce
address
antibodies, you would tend
these
to agree with
also
me.
6 pillars
Although this
is
interferon is
the
not
future and is the
exactly
way
right,
to go, and that's definitely
because we
going to
would rather
improve
say
patient
that
outcomes and our
B cells, and not all B cells, by the way,
also
are rather the precursors of plasma cells that secrete antibodies, and these antibodies may be IgGs, may be autoantibodies involved in myositis. But this is also a bit short, because B cells are also cells that present antigen and interact functionally and in a bidirectional way with T cells. And also they produce cytokines. So B cells are cells that interact with T cells, present antigen, produce cytokine, and are the precursors of plasma cells that secrete antibodies that may be autoantibodies in myositis. So this being said, how can we study this question of the role of B cell? Well, probably we would have to see whether autoantibodies as biomarkers are associated to different forms of myositis. We have to determine whether some of these autoantibodies are more than biomarkers. They could be really directly pathogenic. We would like to see what IgG depletion does therapeutically. we would like to see maybe if B lineage cells are present in the muscle of myositis, and if B-cell depletion could be treatment effective. So I will try to briefly cover some of these aspects. Regarding the first one, it is very clear that autoantibodies are a hallmark of myositis. They are associated to clinical forms, they are associated with prognosis, and I would not go into more detail because this will be the subject of the next talk. Regarding the question of pathogenicity, I will take the example of immune-mediated necrotizing myopathy, which, as you know, is a progressive disease, can be severe. The CK levels are very high, and we see necrosis of myofibers here. And the two main target antigens are the SRP complex and HMGCR, both of which are present in the cytoplasm near or transmembrane of the endoplasmic reticulum. Thanks to the fact that the murine antigens are similar enough to the human antigens, we can show that the human autoantibodies can react to mouse muscle, mouse molecules, whatever. So it was possible a few years ago to determine that if you take a normal mouse, you transiently immunosuppress them and inject these mice with serum, plasma, IgGs from patients, well, you get a reduction in the muscle force, in the grip strength, and this is true for the serum of patients with anti-SRP antibodies or with anti-HMGCR antibodies. This is associated with necrosis, phagocytosis of dead cell by macrophages, and you can count these cells. So this is well known now, and an idea could be that these autoantibodies, well, they recognize something on the muscle cells, probably they activate complement macrophages, they kill cells, and this muscle cell release CK, and this is why CK is high. But this view is not the most current view recently because we have some new data, and it started by this observation by the group of Olivier Benveniste that if you add antibodies from patients on cell cultures, well, you get an effect. I mean, these cells don't like it. And in this situation, you have no complement, no macrophage, no ADCC, nothing. So it's like antibodies could be pathogenic, but maybe involving some kind of direct function that is not triggering other type of immunity. So I would like to remind you, if we take the HMGCR, IMNM, for instance, that HMGCR is an enzyme. So an enzyme will transform, catalyze the transformation of a substrate, HMG-CoA, into a product, mevalonate, that will ultimately give rise to some isoprenoids and cholesterol. So if an antibody blocks this, you will not produce mevalonate, and this is also what statins do, because statins are HMGCA inhibitors. But this happens inside a cell. So if your antibody has to do something like inhibiting HMGCR, well, it has somehow to enter the cell, and this is very exciting research going on about this, because it's quite probably the case. So we took IgGs from patients. This is an enzymatic test of HMGCR function. This curve is the normal curve. It's a degradation of the substrate here. If you can block it with statins. And now if you add IgGs from patients with anti-HMGCR autoantibodies, well, clearly you inhibit this. So this has been observed also by the group of Alain Meyer, with this is the curve on inhibition. And so you see that as compared to controls, there is a reduction of HMGCR activity when you add antibodies from patients. And this is a very nice control. It's human SRP, and SRP is different antigen, same disease, different antigen, no inhibition. So I think it's a very good control. And also they have observed, after electroporating the antibodies directly into the cells, some lipid droplets here that suggest some dysfunction in lipid metabolism. So yes, well, antibodies against HMGCR block this. But in IgG in serum, we're never sure what is the effector mechanism. We could have different antibodies, something. So we wanted to know a little more, and we produced monoclonal antibodies against SRP and against HMGCR. So this is done the very simple, classical way, immunizing mice, getting the spleen cells, producing the hybridomas. But then we sequence the hybridomas and transfer this into a human IgG1 backbone, because IgG1 is the isotype that we find in patients, and we want to have a humanized antibody. And so now the question is that, is this monoclonal antibody inhibiting the enzyme? And if yes, if I inject it into mice, can it enter the cell, block something, and is the mouse sick? And so the answer is yes, we could generate, that was the easy part, monoclonal antibodies that can block HMGCR, but now inject it to mice, and this is I kept this result for this Gcom meeting. Well, we can inhibit the muscle function in mice, and we see images of necrosis and accumulation of macrophages. So these monoclonal antibodies, we know that it binds to the target, and it has to enter the cell in a way or another. But since we have this tool, we are currently investigating how this happens, this entry. And luckily enough, it's the same for anti-SRP. Also, humanized monoclonal antibodies, we just got this result very recently, also inhibit muscle strength and induce some necrosis. So antibodies can be pathogenic in IMNM here. The other point was about depletion of IgG, because if IgG plays a role in myositis, well, if you deplete IgG, you do something for the patient, and there are different ways of doing this. I took this example here, you know, the Fc receptor, FcRn, that protects the circulating antibodies from degradation through this cycle. So if you block the antibody here, the FcRn, sorry, now the circulating IgGs are degraded, and so they are cleared of the circulation. The question was, can this do something in a mouse model? Because now we have a mouse model of pathology, so we injected mice with patients' IgG, wait until they are sick, so it means that the antibodies went to the muscle, did something, and now the question is, if I remove the antibody from the circulation, do they get better? Which implies that you cannot remove the IgG from where they are, because if the mechanism was like, I bind to something, I activate complement, and I kill the cell, well, the treatment would not be effective, right? So it goes with this idea that there is an ongoing inhibition of something. And the result is, yes, we can, at day 7, we inject this drug, efgartigimod, and we eliminate the IgGs, and the specific IgGs here. We show that it helps removing the antibody in the muscle. We can even elute this from the muscle tissue, and it's very low after treatment. And the effect on necrosis is significant. With less necrosis, more regeneration, because there was necrosis before. You relieve this pressure, so now the muscle regenerates. And the more important, of course, is an you recover the grip strength, and so this was also the basis of a clinical trial. The results were presented by Hector Chino recently at ACR, and efgartigimod is having a positive clinical effect in patients. There are other ways than efgartigimod, nipocalimab. There are other inhibitors, and they are currently investigated. So for IMNM, it's certainly a pathway, which tells us that knowing the pathophysiology is also a way to guide therapy. 4, are B cells present in muscle? Well, the B cells, they're produced constantly in the bone marrow. They circulate in lymphoid organs, and when they are activated to plasma cell, the plasma cell home back to the bone marrow to secrete antibodies. But now, can we find B cell in muscle? Well, you probably know that the answer is yes. In adult dermatomyositis, you can find some B cells. Also in JDM, there are forms where you can find more of them, and even up to like really tertiary follicular reaction here in some patients as well. So yes, we can find them in DM, we can find them in the anti-synthesis syndrome, we can find them, quite surprisingly, quite a lot in IBM, which is less antibody-driven form of myositis, and not much in IMNM, probably because the antibodies are produced from the bone marrow. In this work, they took muscle biopsy to perform single nuclei sequence RNA-seq, identifying nicely the B cell and the plasma cell, and IBM was, here again, a bit to my surprise, the more B cell rich form of myositis, more than IMNM, for instance. And very recently, the group of Andy Mammon, and I think, I don't know if Andy is in this room, I think he's coming, did this superb work that you can find the preprint in medRxiv. This was performed in dermatomyositis, and the one associated with anti-Me2 antibodies. And you see the heterogeneity of the muscle when you block. If anti-Me2 cell enter the cell and do something, it will interfere with the NERD pathway. I don't have time to go into details, but it would end up with a specific signature. And what you see here, this beautiful picture, is that you find this pathogenic signature in some cells, but not others. And these ones are perfectly fine. So now what's the consequence in terms of pathology? Well, the consequence is that these colored cells are fine, but these one lack expression of some key structural protein. So in other ways, it's like the antibody enters the cell, does something, we have a signature, and the cell is sick. But what is very exciting here is look what they found. There's a little cell here at the head of this arrow, and this cell is a plasma cell. So B cell can be found, but plasma cell can be found. And what is probably revealed by this work is that plasma cell can locally secrete antibodies. And this is why just locally these cells capture the antibodies. They may even possibly capture the IgG RNA, and then they have this signature and get sick. So B cell can be found in muscle. Plasma cell can be found in myositis muscle. Now, if they play a role, is B cell depletion treatment effective? So we have different ways now of depleting B cell. The first was, of course, rituximab, the anti-CD20 antibody, and now we have different anti-CD20 monoclonal antibodies, OB, OFA, different ones. You can also target with daratumumab the plasmablast and plasma cell. And then we have the CAR T cells that can target CD19 and deplete the B cells, and also the anti-BCMA CAR T cell destroying plasmablast and plasma cells. And we also have some bispecific T-cell engager or bispecific antibodies that can do pretty much the same. They bring together the target B cell or the plasma cell with a T cell, activate it, and it kills. So now we have quite an army of approaches to kill the B cell and try to see what it does in myositis. What are the results of rituximab in myositis? Well, they are a bit, I wouldn't say controversial, but they are variable. There has been this meta-analysis reviewing several studies in more than 300 patients, and the level of complete remission is quite low. I mean, it's 13%, and the partial response, which means that you have no response in 50% of the cases. So, yes, it can help, can do something in combination with other therapy, but it's not the revolutionary therapy, and the new molecules, OB, OFA, OCRE, are, for the moment, we have very few reports, case reports, and the results are improvement in muscle or no improvement. So it's yes or no, we need more time to know more. But the big thing is the CAR T cells. You may know the CAR T cells are a new therapy initially developed for B-cell lymphoma and lymphoblastic leukemia, and now to myeloma as well. It is, the principle is to genetically engineer your own cells so that they express a chimeric receptor that would recognize CD19, for instance, expressed on B cell and less on plasma cell, or BCMA, highly expressed on plasma cell and less on B cell. And when these engineered T cells are given back to the patient, it's an autologous therapy. Well, they would kill the target and activate by activation of this cytotoxic function of these CAR T cells. So it's really burning news, because what I'm going to say today will be outdated next week, because if you look at the literature, you find new papers at a pace that is just incredible. And it's incredible. There's a move of the CAR T cell research from hematological malignancy to autoimmune diseases, and among these autoimmune diseases, you have lupus, you have systemic sclerosis, and myositis is really a big issue. So the first report was by this German group of Georg. You see a patient. This is the CK value of this patient with an anti-synthesis syndrome. Rituximab has an effect, but it's transient with a relapse, and then rituximab loses effect. You have to add several lines of therapy, and now when you treat with CAR T cell, you have normalization of CK, the muscle force, the patient gets better, and the autoantibody is no more detectable at a time where the B cells are back, by the way. A few other patients, a new patient from this group with anti-Jo1, but also an anti-PL7, anti-synthesis syndrome, and with good result here, the CK are very low. Many different reports from now regarding anti-CD19 or anti-BCMA cells targeting different forms, and the result is drug-free remission, drug-free remission, drug-free remission, improvement in all domains. So it looks like it's pretty efficient, although I cannot say it occurs in every patient. It's not the case, and you have some relapses, and we need more results to be, we should be cautious about this, because this is a low number of patients. It happens also, it seems to be efficacious in JDM, as far as it was tested in this report here. Also on extramuscular symptoms, like in the lung, in this MDA5, anti-MDA5 positive dermatomyositis. As you see, the patient is here drug-free after CAR T cell therapy, and at a time where the B cells are back. And this is important. I will come back to this point briefly. For IMNM, of course, the autoantibody-driven disease, it looks like it works. You see this very important B cell depletion here, but rapidly the B cells are back, and they are back more rapidly than after lymphoma or leukemia. Nobody really knows why, and it's interesting to think about this. But at the time where the B cells are back, the autoantibodies are not, and so this is a log scale, so they are still very low. And interestingly, in this study, the vaccine antibodies are still here because you killed the CD19 B cell, and probably the long-lived plasma cell, those secreting the anti-vaccine antibodies, they are still around. So that's interesting. What about anti-BCMA CAR T cell? Well, in IMNM, we also have one case report here, one case report there, but it seems to work as well. You see the anti-SRP curve. It's like negative. The CAR T cell, they expand, and then they disappear, and the CK level, of course, go back. This is autologous, so it's very complicated, very expensive, cumbersome. You have to take the cell to send them to, it's big pharma generally, or in the German case, or in Spain or some countries, you can do this academically, as we say, in your hospital, but then they have to be frozen, sent back, and so on. So it's complicated, and we would like to have ready off-the-shelf CAR T-cell, but they would be allogenic. And in myositis was one of the first cases, it's a Cell paper, where they studied the effect of allogenic anti-CD19. The problem with allogenic is that the immune system will get rid of the CAR T-cell. So if you have cancer, you don't want that. But for autoimmunity, you just want to reset your B-cell system. So maybe it's going to be an effective therapy. We need to know more. But in this case, look, you have one case of IMNM, and so that's interesting. The CAR T-cell, they expand, they go back, the B-cell are 0, but then they come back. But when they come back, the entire SRP are still at 0. The patient is getting better. The MRI is much better, and even the biopsy looks subnormal. So we have to understand something here. Rituximab, if you may have experience with rituximab, when you use rituximab and you look at the B-cell in the blood, it's like 0. After a few days, 0, like 0. So why, if a treatment that brings your B-cell to 0 fails, another treatment that takes your B-cell to 0 would succeed? And so we understand this now. First, it's because the rituximab or anti-CD20 depletes this in the B-cell development. CD19 CAR T-cell depletes a bit earlier, a bit later, but the main reason is probably not that. Look at these pictures in the lymph node. It's that after rituximab, you have 0 B-cell in the blood, but there remain B-cell in the lymphoid organ, and this is probably the same in the tissue. Whereas after CAR T-cell, you completely clear off. These CAR T-cell, they are T-cell, they enter the tissues, they enter the lymphoid organs, it's their job, they kill their target. And so when the B-cell come back, there are new B-cells from the bone marrow, and they are not exposed to maybe environmental trigger that have triggered the disease. So that's probably why the disease doesn't come back. And I will finish very rapidly now. You have autoimmune B cells. They are destroyed by the expansion of this CAR T cell, and the naive B cell that come back, they are not the same B cell. It's not recurrence from a pool of B cells that's here, is that you've really cleared them. So it looks like a nice story, but there can be difficulties. I also want to stress this. Look at this patient. It's very interesting. It's a patient with an anti-JO1 myositis, and he received CAR T cells. It's the blue line, so the CAR T cell expanded and then, disappeared as usual, and the disease got better, but then there was a recurrence. So this group, what they did, they tried to re-inject CAR T cells, and this had 0 effect. The CAR T cell did not expand, the CK went up, it was total failure. So the reason what can happen, and what can happen is that the CAR is chimeric, it's in its name, it's foreign, it's non-self. So the patient rejected the CAR T cell by an immune response against the CAR. And this is something you don't see, as far as I know, in hematological malignancy, but in this autoimmune background, it's possible that we're going to see this. And they did something which I think is quite clever, because you have anti-CD19 drugs, so they rescued this patient by using another CAR. It's also autologous, but now they use an anti-BCMA, the patient is fine and has, with this therapy. I will not spend too much time, but you can. There was a hope that we could do something simpler than CAR T-cell. It's mobilizing the own T-cell with a T-cell engager against CD19, like blinatumomab, and it kind of works, but you need to inject rituximab at the same time. You need several injections. It's not like the CAR T-cells. So we cannot draw definitive conclusion, but probably it is less effective. Here you have the first case published very recently of a T-cell engager that mimics the function of anti-BCMA CAR T-cell. It goes teclistamab, and in 2 patients, well, one was a non-responder and the other was a responder, so it seems to be also a variable result. We need more. So to conclude now, do we have association of the disease and autoantibodies? Well, the answer is yes. Are the antibody pathogenic? Well, in many instances they are. Is IgG depletion treatment effective? It is. Is B-cell depletion treatment effective? It is. Is B-cell depletion, do we find B-cell in the muscle? Yes. Is depletion effective? Well, often. And so I think it's reasonable to conclude that B-cell, B-lineage cell, and autoantibodies have a role in myositis. And CAR T-cell hold promises, but long-term safety need to be assessed and long-term efficacy as well. So it's just the beginning of the story. So I will thank the people in my lab, Catalina Abad in this room, who did part of this work with Laurent Drouot and other people, a long-term collaboration with Olivier Baveniste and Yves Allenbach on all this work, and also with Alain Meyer and Margarita. With the permission of Lucy, notably, I just tell one thing, is that myos has just joined this nice federation of FOCIS, the Federation of Clinical Immunology Society. And so for this, there will be this meeting in June, and we will organize, this association will organize a special symposium during this meeting with these people, including myself. So if you're interested, you can go.
So, thank you. We have time for one quick question. Okay. If nobody is asking, I will also... oh, okay, sorry.
Hi, interesting talk. So I would like to listen to your thoughts on the effectiveness of the B-cell inhibitory therapy in those who are antibody negative. There would be patients who are antibody negative, and as you have elucidated that B-cell is the pathogenic marker, in that case, how to take the effectiveness of anti-B-cell therapy in the patients who are antibody negative?
Excuse me, I'd like to make sure, how the antibody disappear, or what is the efficacy of CAR T-cell in antibody negative?
MSA negative.
Yeah. Yes, well, B-cell produce antibodies, but B-cell produce cytokine, B-cell talk to T-cell. We have diseases where CAR T-cell, depleting B-cell, is effective, whereas the antibody probably don't play a major role, like multiple sclerosis. So I think that depleting B-cells has a role independent of the pathogenicity of antibodies.
Okay, thank you very much.
So next, we're going to invite our next speaker, Thomas Koo, who is a rheumatologist in South Australia, working across Adelaide Health Networks. And his focus of his research is in muscle disease, including clinical outcomes and health experience of the Aboriginal and Torres Strait Islander people. Thomas.
Thank you, and thank you very much for the introduction. I'll start by apologizing. I've managed to pick up a cold somewhere between Adelaide and here, so I lost my voice. It's come back today, and I hope it lasts for the next half an hour. These are my disclosures. So when I was asked to give this talk, I was asked to speak on the clinical associations of different autoantibodies. And having now finished the talk and put all these slides together, I've discovered that what I've actually presented is a love letter to autoantibodies. And I think all of us can safely say that we do have a very special place in our hearts for myositis antibodies and the utility that they've had for our clinical practice, as well as understanding better how these diseases come about. And so as part of this love letter, if you will indulge me, I thought it is very worthwhile taking a short step back in history just to recognize how recently all of these antibodies have come into our lives. And so if anyone was watching Jaws in 1975, you were watching it in a time when there were in fact no myositis-specific antibodies published in the literature. In 5 years later, if you were watching The Empire Strikes Back, there was but one. And little did you know that 3 months later, Anti-Jo1 would be published, and we know what a big effect Anti-Jo1 has had on our understanding of myositis. Skip forward 10 years, a further six antibodies have been discovered, 5 of which were anti-synthetase antibodies. And then another 10 years later, if you're watching Gladiator, only 2 antibodies had been discovered in the intervening decade. And then the penultimate Harry Potter movie, a further 6 antibodies. If you go from 2010 to 2020, the first thing we recognize is that the COVID pandemic meant that no good movies were released in 2020. And in fact, no antibodies were published between 2010 and 2020. And I think reflecting on the history of myositis antibodies, we recognize that there has been a certain chaos to the discovery of these antibodies, but also a sort of driving purpose in terms of trying to understand good biomarkers for these diseases and good ways of identifying diagnostic tools. And so in this chart here, you can see above the green line the antibody discovery over the years, and then below the green line the very assiduous efforts to try and understand better how to classify conditions which are probably different but are unified by the fact that they can affect muscle as one of their manifestations. And indeed, nothing goes towards the chaos of antibody discovery more than the nomenclature that we use. And in fact, as you can see, many of them are named after their molecular targets, but quite a few of them are named after other places. And for example, the latest anti-synthetase antibody, anti-Li, was named after the location of its discovery. If anyone here knows where anti-OJ and EJ came from, I'd love to speak to you. I've dived into the literature and never quite worked out exactly where they came from. Moving on. So moving on to more clinically relevant topics, the question of the clinical associations of different autoantibodies really boils down to the question of how can antibodies help us in our everyday clinical practice. And so I think they can help us in a few different ways. Antibodies are obviously helpful with diagnosis. They can help guide screening, particularly in the early phases of disease. They may help us prognosticate conditions such as myositis. They may potentially predict treatment response, give us some idea about monitoring, and maybe one day even give us some insights into how we might prevent the onset of inflammatory muscle disease. So what I thought I'd do is I'd talk about each of these phases in turn rather than going through each of the antibodies individually. And I think it's also important to note that I won't talk about how these antibodies are tested for, because we know that the testing methods and the reliability of those methods are obviously key to how we interpret a positive and a negative result. So let's move on to antibodies and diagnosis. We know that in medicine and in rheumatology in particular, we do a lot of pattern recognition. The patterns that we put together often revolve around things like the history taking that we use, different features of the physical examination. In muscle diseases, we know that neurophysiology and muscle biopsy came into the armamentarium actually at about the same time. And then more recently, imaging has also provided very useful techniques in order to work out the patterns that we discern in myositis as well. And then over the last 50 years, we recognize antibodies as being key to understanding and trying to tie together the overall picture of what we're trying to look at when we look at these patterns. And I think one of the things that first drew me to rheumatology and to myositis is this concept of a myositis antibody and a clinical phenotype, and the fact that these tiny little antibodies could have such distinct and clear and stereotyped clinical manifestations. And I think a very good example of this is the anti-synthetase syndrome. There are 20 anti-synthetase antibodies, and 9 of them have been reported well. And we recognize that in addition to inflammatory muscle disease, anti-synthetase syndrome can obviously also comprise inflammatory skin manifestations, inflammatory arthritis, the Raynaud's phenomenon, and nail-fold capillary abnormalities, mechanics hands, and interstitial lung disease. And uniquely, it is these extramuscular manifestations which do vary subtly between the different anti-synthetase antibodies, and are in fact also good differentiator factors between different antibody groups. So for example, in contrast to anti-synthetase syndrome, which can comprise all of these manifestations, when you have a condition like anti-SRP, immune-mediated necrotizing myopathy, it is essentially a disease of muscle, with very few, if any, extramuscular manifestations. And these patients can certainly have very prolific muscle involvement, strong axial muscle involvement, dysphagia, and very, very high CK levels. Importantly, it's not just the myositis-specific antibodies that have unique clinical complexes. For example, anti-PMSCL or anti-exosome, although we think of it as a myositis-associated antibody, appears to have a quite clear clinical phenotype as well, where they have a higher rate of dropped head and bent spine syndrome and neck involvement. And in this fluoroscopy image here, you can see a patient who was simply unable to extend her neck, but also having esophageal dysmotility, which is potentially one of the more sclerodermatous phases of this disease. They can also have unusual skin rashes that can resemble psoriasiform rashes, sclerodactyly, and Raynaud's phenomenon and digital ulceration, for example. And so I think one of the interesting progresses in myositis antibody research over the last decade or so has been not just that there are unique clinical phenotypes, but in fact that for some of these antibodies there are multiple unique clinical phenotypes, and they are still stereotyped and still clinically concordant, but there are different clusters, I suppose, that associate with each of these antibodies. And I think a good example of this is an antibody which has certainly captured the literature and has resulted in 10 times the publication of any other antibody lately, which is anti-MDA5 dermatomyositis. And so in this chart here, you can see the vast spectrum of manifestations that this particular autoantibody can be associated with, but not all patients develop all of these manifestations. And there have been great work put towards trying to cluster patients to determine different phenotypic clusters and why that might occur. And some of this work, for example, has been done in the French myositis cohort, where they've described a rapidly progressive interstitial lung disease cluster with very high early mortality, common mechanics hands, and mild elevations in CK, a rheumatic cluster where arthritis is common, IOD is common, but RP-IOD is uncommon, and then a vasculopathic cluster where the skin ulceration, digital necrosis is very common, CK elevation is much more common, but interstitial lung disease is less common. And many groups around the world have tried to establish phenotypic clusters as well as clusters according to biomarkers, and I think it's still an area of very, very prolific research in terms of better understanding why specific patients with anti-MDA5 develop specific manifestations from that slide I showed earlier. One of the other antibodies which has also shown this unique phenotypic heterogeneity is anti-HMGCR. So we recognize that most patients with anti-HMGCR are the patient shown on the screen, an adult who is statin exposed for a few years, who then develops a subacute onset of proximal muscle weakness and typically has very minimal, if any, extramuscular disease. However, we also have reports of patients who have this sort of chronic limb-girdle muscular dystrophy-like phenotype, and that can occur in adults as well as in children. We know that children can be affected by anti-HMGCR. They are universally statin naive, but they seem to have more severe, more refractory disease, and also appear to have more extramuscular manifestations, particularly rashes. And then there are patients who are statin naive and adults, and these are patients who are more commonly reported in Asian ethnicities, female more than male, and a younger group, obviously given that they're not exposed to statins. And then finally, there's a group which is not antibody positive at all, but has a unique phenotypic similarity, which are patients who have gene variants of the HMGCR gene itself. And these patients appear to have a more limb-girdle muscular dystrophy type phenotype similar to the chronic myopathy that I spoke about earlier. But what I think is most interesting about this is not the fact that there are these different phenotypic variants associated with an antibody or a gene variant, but the fact that there are probably overlaps here which we need to investigate further. And better understanding the genetic environmental factors which fall into these highlighted areas of overlap, I think could really help us understand exactly what the anti-HMGCR antibody is doing and how it culminates in disease of different phenotype despite the sort of mantra of a single antibody and a single clinical phenotype. So moving on from diagnosis, we'll talk a bit about screening. And I think probably the biggest progress in screening over the last few years has been the understanding of cancer screening in myositis. By Alex Oldroyd and the cancer screening IMAX group, the guidelines suggest that antibodies are key to our understanding of how to screen these patients. But antibodies are clearly not the whole story here. So, for example, TIF1 gamma and NXP2, which we know are the antibodies most associated with malignancy, when TIF1 gamma was looked at in UK MyNet, there's this very clear delineation according to age. of cancer incidence with anti-TIF1 gamma. In this particular cohort, it was around the age of 40, and you can see that below the age of 40, in fact, very few of these patients with anti-TIF1 gamma, none of them actually developed a malignancy. And I suppose this is in keeping with our understanding of juvenile idiopathic inflammatory myopathies as well, where we know that NXP2 and TIF1 gamma are the most commonly represented myositis-specific antibodies, but also know that malignancy in children is essentially unheard of and limited to sort of case reports with inflammatory muscle diseases. In addition, a few years ago, there were reports of anti-CCAR1 and anti-SP4, which are these interesting antibodies that are neither myositis-specific nor myositis-associated, but potentially modulate the effect of anti-TIF1 gamma. And so anti-CCAR1 was discovered when anti-TIF1 gamma and cancer was analyzed. And in the green, you can see that these are patients who did not develop cancer, and they were patients who had not only got anti-TIF1 gamma, but also prolific other autoantibody production. Whereas in the blue, these are patients who developed cancer very closely in proximity to their diagnosis of dermatomyositis, and these are patients who typically only had anti-TIF1 gamma and a very limited spectrum of antibodies. One of the antibodies that was identified in that green band is this anti-CCAR1 antibody, and when it was analyzed in terms of CCAR1 positivity and TIF1 gamma positivity, these are patients who had a later onset of cancer, unlike on the right, where they were mostly clustered in that first 3 years. There was a more even distribution in the CCAR1 positive patients, but interestingly, they also had earlier TNM staging of cancer. And so they didn't have cancers which had spread or had grown as much as patients who were CCAR1 negative. In a similar fashion, anti-SP4 was also reported in the same year. And like CCAR1, this appears to be an antibody that only exists in patients who have anti-TIF1 gamma. And in both the discovery and the validation cohort, again, very low rates of cancer were found in the patients who were anti-SP4 and anti-TIF1 gamma positive. Now, I recognize that CCAR1 and SP4 have not had this data replicated outside of the USA so far, but I think it's a really interesting insight into the fact that there is this interaction between antibodies, muscle, skin, and cancer, and it shows that the sort of interactions of the immune system in developing malignancy in these autoimmune myopathies is really an area of very, very interesting research moving forward. So we've spoken about diagnosis and screening, but can these antibodies help prognosticate? And I think that's a tricky question. What I'm going to do on this slide is a bit heretical, so I apologize if I offend anyone in advance. But if we try and graph out mortality and relapse risk of different antibodies, there are a few publications in the last few years which have looked at this. And so in a group of patients with interstitial lung disease, according to myositis-specific antibody, Jennifer Hanna had a look at whether different antibodies had different mortality. And the overall finding was that from these 3 hospital networks in London, anti-PL7 had the highest hazard ratio, although anti-MDA5, as you would expect, within the first 3 months of diagnosis, had the greatest hazard ratio. Although when patients survived that period, the hazard ratio was significantly improved. And part of that improvement was the fact that anti-MDA5 generally has a very low relapse risk once patients get past their initial inflammatory phase. Anti-PL12 and anti-Jo1 appear to have a lower all-cause mortality compared to anti-PL7. And then in a different publication, having a look at anti-synthetase syndrome versus anti-MDA5. Again, it was concluded that yes, anti-MDA5 has this very poor early prognosis, but anti-synthetase syndrome has a much more frequent long-term recurrence than anti-MDA5. If we have a look at radiological studies of anti-synthetase syndrome ILD versus anti-MDA5, it suggests that anti-synthetase syndrome has more persistent ground glass opacities than the more fibrotic phenotype of anti-MDA5 ILD. And so potentially, if we add a third dimension to this graph, anti-synthetase antibodies are more associated with persistent disease as opposed to anti-MDA5. If you extend this further and have a look at non-ILD associated antibodies, there is some evidence that anti-SRP and HMGCR have a better overall survival than patients who have interstitial lung disease. And we know that these antibodies are associated with immune-mediated necrotizing myopathy with a very high risk of relapse as well as persistent disease and CKs which often don't go back to normal. And then recently, a publication has looked at comparing SRP with anti-HMGCR and suggests that anti-SRP has more ongoing disease than anti-HMGCR. So I guess what I wanted to show with this slide is not so much the comparative scale of these different antibodies, but the fact that the landscape of myositis antibodies does have prognostic benefit. And hopefully at some point in the future, when we synthesize all of this information, we can sort of plot on this area where a certain patient with a certain antibody sits and recognize the different sort of spatial areas of this graph where different antibodies reside. So moving on from prognosis, can antibodies predict treatment response? And here we start to get into slightly more uncertain territory. So we all know and have great affection for the RIM trial published in 2014. And in the post-hoc analysis of the RIM trial, it was recognized that patients who were positive for Jo1 and Me2 antibodies appeared to respond better to rituximab than those who did not have these antibodies or who were antibody negative, according to the antibodies at the time. And so potentially, there are different antibody subsets which respond better to different therapies, although there is still a lot more work that needs to be done in this sphere. Another example is anti-HMGCR. We know that clinically, anti-HMGCR immune-mediated necrotizing myopathy appears to respond exquisitely well to intravenous immunoglobulin. And indeed, in this recent trial, when monotherapy IVIG was compared to non-IVIG therapy-based regimens, at 6 months, IVIG had a better improvement in normalizing CK, and indeed all of these patients improved on monotherapy IVIG in terms of improving their CK on the left and improving their muscle power testing on the right. And so I guess it suggests that different antibodies may well respond to different therapies. And so I wanted to try and put together anti-MDA5, and I stopped halfway through because it just got too confusing. But I think we still, for example, with anti-MDA5 and rapidly progressive interstitial lung disease, have a way to go in terms of determining exactly what the best therapies are. And although we recognize that potentially combination therapy is more appropriate than step-up therapy in anti-MDA5 dermatomyositis, we don't yet have a good understanding of exactly what therapies should be targeted to this particular antibody subgroup. And I think that means that what we don't yet have, but which we are definitely moving towards, but particularly having heard the last three speakers, is this concept of a myositis-specific antibody, and from that inferring a specific pathophysiology, and from that then determining a targeted therapeutic. And I think this would potentially be the holy grail of understanding how we treat different autoantibody subgroups in inflammatory myopathies. So what about monitoring? Now, Olivia spoke a bit about pathogenic antibodies and what we think of as being pathogenic and not, and I know that the boundaries of that are starting to be pushed a little in the understanding that there are some antibodies which you did not typically think of pathogenic, but which do have an effect on the antigen that they bind to. But in terms of monitoring, I guess the ones that spring to mind are anti-MDA5. In one of the early publications of anti-MDA5, it was recognized that in patients who become inactive, their anti-MDA5 titer does seem to go negative, although this has not been reflected in all the cohorts that have been published since. And then I suppose in a small cohort, anti-HMGCR also appears to potentially have monitoring value in that patients with anti-HMGCR IMNM, where their antibody becomes undetectable, were more likely to have a sustained clinical improvement without relapse compared to those who did not have an anti-HMGCR that became negative after treatment. But there is still a large question mark over how we use antibodies and what test is most appropriate to test these antibodies in order to monitor progression and monitor for response to treatment. So the last thing I wanted to touch on is the idea of prevention. And I think we all know that there is this clinically evident phase of muscle disease which is preceded by a preclinical phase of autoantibody positivity. And then, I know I've only done 2 lines here, but many, many different genetic and environmental risks that culminate in the development of autoantibody production. And it is this phase before clinically evident disease that offers an opportunity for prevention. And so this has been explored in, say, rheumatoid arthritis, where some of my Australian colleagues have published 11 ways to avoid rheumatoid. And some of these may be extrapolatable to myositis, for example. But I think what we have for some forms of myositis is a very unique situation. So, for example, if we take anti-HMGCR, we know that there is this very clearly defined environmental risk of statin exposure, and we know that there are well-reported genetic risk factors, for example, HLA-DRB1 1101. And it raises the question, if we were able to identify with accuracy the genetic risks that predispose statin users to develop anti-HMGCR, Will there ever come a point in time where we take that risk, apply it to a patient before prescribing them a statin? I don't know the answer to that. I think it's a difficult question, particularly since how prevalent statin prescriptions are. But I think it gives a good example of an opportunity to try and avoid the development of an antibody and avoid the development of a clinically relevant disease if we are able to identify risk factors for the development of these conditions early. So this is what I've spoken about today. I've spoken about the different clinical relevance of antibodies for each of these phases of the patient journey. We recognize that it can be helpful in diagnosis, definitely. It helps guide screening, which we have good evidence for. Potentially, it helps prognosticate, predict treatment responses and monitoring, and maybe one day in future we'll have a time when we can use antibody positivity preclinically to try and prevent the development of disease. But having recognized the distance we have come with myositis antibodies over a relatively small period of time, I think this is a very exciting meeting for me to try and appreciate more the forefront of research. And I think looking at a Lisbon landscape of myositis, we've been so lucky to have a time when, from a time when myositis was thought to be an infection, we then discovered that myositis was actually polymyositis and dermatomyositis. And over the years, we've had inclusion body myositis, immune-mediated necrotizing myopathy, and overlap myositis, making polymyositis a progressively smaller domain of the landscape. And these days, we recognize that these are actually probably separate diseases. And in fact, the histology would suggest that there are different pathological processes occurring in each of these myositis subgroups. But more recently, we know that antibodies have become the main driver in terms of subgrouping these diseases. The anti-synthetase antibodies, 5 dermatomyositis antibodies, 2 IMNM, and then the myositis-associated antibodies of IBM and overlap myositis. And the greatest benefit, I think, of these clinically is the understanding that they have these unique clinical manifestations which associate not only with subgroup, but also with the different antibodies that we now can routinely test in each of these patients. So thank you very much to everyone, and I also appreciate all my friends, colleagues, and collaborators over the years. If you see your face up there, I'm very, very grateful for having been able to have the chance to get to know you and work with you. So thank you very much.
Thank you very much for this beautiful talk. Please don't be shy. Wave if you would like to ask a question and someone will come to you. Tony.
Love that talk. And I understand, as somebody who is probably in the jaws, you know, when I grew up, we didn't have maybe Joe. And so I love how the field has progressed. So I'm a neurologist who always believed in biopsy, 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 and poo-pooed the antibody, but you've made me a believer over the last 3 decades, and I think that's great for the field. I do fewer, but I think the biopsies are interesting. But, you know, I also did the polymyositis as a unicorn dragon editorial that got poo-pooed, but I also like to see how that has narrowed in its... So I want a question about what is anti-coup? Because Werner Stenzel had a beautiful paper like a year or so ago, and the biopsies looked indistinguishable from inclusion body myositis, and the patients even had IBM. And I just had a patient recently who, older lady, can't flex her fingers, biopsy looks IBM, you would just say, meets any criteria of IBM, but the coup was positive, and the only weird thing was her CK was like 3,000. So does coup not belong in poly? Does it belong in the IBM thing, or is it something that can have, you said, different phenotypes? I was just wondering what people's experience, because that's perplexed me, or anybody's had similar experience.
Thank you. I'd love to do some research in anti-coup at some point, mostly because of its similarity with my surname. But I think we think of it as a myositis-associated antibody, and I think probably the differentiation of myositis-specific and myositis-associated antibodies does it's probably something that we need to move away from a little bit, because I think there are unique associations of associated antibodies as well. And as I described with PMSCL, there are specific clinical phenotypes that can be described, as well as histological appearances that associate with those antibodies. So I think the rarer autoantibodies that we consider, things like anti-COO, certainly need a lot more research and understanding of how they manifest and the clinical patterns that they associate with.
I think we have a question here. Please, go ahead.
Yeah, thank you. Keith March from Gainesville, Florida. Beautiful talk. My question for you is, what are your thoughts about developing assays that might be patient-specific functionally, not only in terms of describing the antibody, but perhaps in taking peripheral blood mononuclear cells from a particular patient and stimulating them with the autoantigens? Perhaps a panel of autoantigens, but I presume you'd probably start with the autoantigen for which you already have the antibody. Do you think we have the biochemical purity of autoantigens to actually undertake such assays?
Wow, big question.
I'm not sure. I think, look, it boils down to the whole question of reliability of assay and what test we use most for these antibodies. You know, the gold standard of immunoprecipitation, we know there are deficits with that. And so I think we need reliable methods of testing. And I don't think we have that for every single antibody thus far. So I think, you know, I guess the overarching answer to your question is hopefully one day in the future we have reliable, specific, antibody-specific and individual-specific or potentially even, you know, specific to certain ethnic groups where we have reliable testing that we can use. For example, I guess one of my particular research interests is the Aboriginal and Torres Strait Islander peoples in Australia who universally have hypergammaglobulinemia. And so testing these patients often results in many false positives on panels that we commonly use. And I think we need more understanding of local laboratory cutoffs, but also cutoffs that are tailored to patients with different ethnicities and genetics, for example. So I hope what you're saying comes true, and I think we come to a point where we have the precision of the technology to deliver that sort of antibody testing for individuals.
I'm sure it's a good discussion later in the meeting. Thank you. One last very quick question, very quick answer, please.
Yeah. Thank you very much. Excluding the pitfalls and limitations of the current screening methods, what is your opinion about the seronegative myositis, in particular the polymyositis phenotype? Do they exist?
Does it exist?
I think we've had a history where we've proven that seronegativity has been progressively eaten away. I think there are more antibodies to discover and define, and potentially antibodies outside of the binary of myositis-specific and myositis-associated antibodies. I guess there will probably always be this group of seronegative conditions where potentially they're not autoimmune at all, but have phenotypes which are very similar to the inflammatory myopathies as we understand them. But I think increasingly what history has shown us and continues to happen is that the group of seronegative inflammatory myopathy, or so-called polymyositis, is dwindling to a sort of mythical beast.
Okay, thank you very much. Very stimulating talk. Don't go away. Don't go away, because now we have 3 short abstracts selected from the very many abstracts submitted to this meeting. So I'm going to pass back to my co-chair to introduce the first short oral speaker.
So we are going to call Linda Chenin. Linda is from Sorbonne in Paris. Oh, okay. Okay. You're attacking from another side. Yeah, welcome.
There they are.
It's a work that is in collaboration with Paris, Lyon, and Lisbon also. So it is titled Interferon Signaling Disrupts Tissue Remodeling. Well, you can see there already. And so we skip this. And please, go ahead.
Yeah. So Linda couldn't make it here today, so I will be presenting instead the work that she carried in the lab in the recent years. So to go forward, as everybody knows, and it was already mentioned several times today, dermatomyositis is an autoimmune inflammatory disease that showcases perivascular atrophy and myofiber damage. And one of the hallmarks would be type 1 interferon present in the tissue, and as it was shown before, it's the most type 1 interferon myositis that we currently know. We wanted to take an approach mainly on the skeletal myofiber and on the effect of the interferon directly on the myofibers. Just to go back to the state of different skeletal muscle fibers, we have 3 subtypes of fibers in the muscle, mainly type 1, type 2A, and type 2X, which goes from slow to very fast. And all of them have one myosin as a marker. And a lot of literature has shown that there is some hybrid fibers that showcase multiple myosin, different ones in one fiber. A lot of recent works have shown that different skeletal muscle fibers have different sensitivity to stress. It was already told before that they can have different sensitivity to metabolic stress, but the thing is, the faster a fiber is, the faster it tends to be fragile, to die in the muscle. And this, we don't know what are the peculiar effects in every myositis on these myofibers. So the deficit is partially explained, of course, in DM by muscle lesion, but there is a lot of deficit, and we wonder if there was an effect on the fiber type repartition directly. And we wanted to phenotype deeply the interferon effects on the myofiber and on the myogenesis. So we went to spatial transcriptomics to investigate the changes in situ at first. So spatial transcriptomics is a method that has been around for a few years now. You do transcriptomics at something like 50 micron levels domains on the tissue biopsy directly. You put your biopsy on a specific slide, you do HN staining or immunofluorescent staining, and then you go through RNA-seq, classical one, to the end to get the transcriptomic profile of every one of your domains. Here what we had was 4 control for healthy donors muscle compared to 5 dermatomyositis muscle. All of our dermatomyositis muscle were MI2 antibody myositis. And when you look at the hematoxylineosin staining, of course, you see the inflammation in the dermatomyositis. You can even see a little the perivascular atrophy from there. And if you go through with what is the interest of the transcriptomic is to see what niche showcase what identity, to tell it simply. We identify something like a little less than 10 clusters, and most of them are characterized by the type of the fiber in the cluster directly. And in addition, you get on the DM side the inflamed fibers that are mostly present only in dermatomyositis condition. And the main change that you can see when you compare control and DM in terms of cluster repartition for the myofiber part is that you almost completely lose your type 2X to A fibers. You lose a little of your 2A fibers only, which are the cluster on the top here. And on the other side, you have less type 1 fibers normal, but you have a lot of type 1 and regenerating. So you have a lot of regeneration with markers of type 1 fibers. So this huge loss of type 2X fibers, we wanted to prove it as a protein level. So Linda did some immunofluorescence here on the left with control and on the right for dermatomyositis. And it's not easily seeable there because it's a lot of purplish views, but if you look at the repartition of the myofibers in control compared to dermatomyositis, you see that you almost lose completely, here you lose completely the myosin 1 expression, so the fastest fibers, the 2X myofibers, and you get a little more of hybrid myofibers between slow and fast types, so myosin 2, myosin 7 subtypes. So first, we have a strong fiber type remodeling, almost an extension of type 2X fibers and a hybrid enrichment. This phenotype has also been shown in other works, notably on aging, notably on IBM. We talk for several years of the fragility of type 2 and 2X fibers mainly. And in our extended data sets, we see that actually it's a spectrum because as you can see on the figure, on control, you have the most of every myosin, but you lose the most myosin heavy chain 1 for dermatomyositis compared to the other myositis. Here we have 5 per condition. So the thing is, what makes dermatomyositis more prone to losing 2X myofibers compared to the other myositis? And in this case, we looked at it still on the transcriptomic profiles. We identified each niche for 2X, 2A, and type 1, so slow, fast, very fast fibers. And we looked at the upregulated and downregulated genes of each of these domains. And what we see that is interesting mainly is that for the slow fibers, the main signals is a downregulation of oxidative phosphorylation directly in the slow fibers, while in the fast fibers, be it 2A or 2X, you get an upregulation of interferon response. So you have much more sensitivity to interferon in the 2X and 2A myofibers in the tissue in situ directly. And going to culture, cellular culture, satellite cells were cultivated, so LC donor satellite cells were cultivated with interferon and with poly-IC, as well as in a control condition, and we went to bulk transcriptomics. And the main results that we wanted to show is putting only an interferon on muscular satellite cells. Actually, you recapitulate a big part of the transcriptome of the dermatomyositis that you get in situ in spatial transcriptomics. So you get upregulation, of course, of interferon response genes. That's normal. But you also get upregulation of slow type myosin, and you get downregulation of myosin heavy chain 1 as the biggest gene, as well as the downregulation of oxidative phosphorylation. And the little plot on the side is showing which part of the genes are common to both datasets. So for the upregulation, you get a quarter of the transcriptome of in situ dermatomyositis with just interferon on satellite cells. So interferon clearly induces a big remodeling of fiber type, which is a new thing, this very strong orientation to one fiber type only with interferon. And in addition, dermatomyositis muscle phenotype is recapitulated only with interferon on myoblasts. We wonder if there was a long-term effect because there have been some cues about senescence in myoblasts from dermatomyositis. So what Linda did was check for the proliferative function of satellite cells of dermatomyositis, and she shows that you lose some proliferative function when you get myoblasts from a DM compared to myoblasts from a control. So inherently you have a difference, but you have no telomere lowering, so it's not a replicative senescence that's shown here. But what you can see is that also you have bigger nuclei, which can also be in some points linked to immunologic toxic senescence. And you have a relocalization of ERA at the PML nuclei level, basically in dermatomyositis myoblasts compared to control, but you can recapitulate a part of this by putting, again, interferon directly on it. So you get back to very, very high relocation of IRA. which means, in simple terms, that myoblasts after 10s, 20s, 30 replication cycles still have some effect of interferon toxic effect on the muscular cells in a very, very long-term way, which is a strong cue on the impact of the interferon myofibers in the study. Sorry. In conclusion, spatial transcriptomics enables to do the study of fiber types with this domain study in dermatomyositis describing that enhanced interferon in fast-type fibers and loss of 2X fibers are specific to dermatomyositis. There is a qualitative phenotype in dermatomyositis that can be recapitulated by interferon stimulation in myoblast culture. And if you isolate myoblasts from dermatomyositis muscle, you show long-lasting effects linked to interferon that you can recapitulate if you put interferon on healthy donor myoblasts. So I'd like to, of course, thank everyone. I want to thank Linda for permitting me to present the results while she couldn't be there. And I want to thank you, of course, for your attention. Sorry for the answer.
So we have now limited time. So if we have one question that has to be brief, we have it. If not, okay. So quick question.
Excellent work. It's not working. Excellent work. Quick question. Do you think the type 2B fibers are atrophying, just a matter of size, or you have less type 2B fibers, like they're just dropping out and they're not there?
There is clearly less fibers. They are probably, a lot of them tend to die faster or easily compared to the other subtypes. So there is clearly less, and it's not just atrophy.
So thank you very much. Thank you very much.
So now we're going to welcome Thorj Dorbentin from the University of Heidelberg to talk to us about immune fiber signaling networks in IBM.
Thank you. Do we have a pointer here? Yes, we do. Perfect. Loving these pointers. Thanks for the kind introduction. I'm Thorj Dorbentin from the Medical Faculty Mannheim at Heidelberg University, and I'm representing the Schirmer Lab today. Thanks again also for hosting us at this beautiful and, from my brief experience so far, also very delicious city. Pistache de Behang, wonderful pastry you have here. And so today I want to present to you cell-cell communication signaling networks in myositis, especially in inclusion body myositis. I declare no competing interest, to the best of my knowledge. So starting with what data we are actually looking at, I'm going to present published data from previously published by colleagues of mine from our lab, led by Sven Wisniewski, where they looked at muscle biopsies from patients of healthy tissue, necrotizing myopathy, and inclusion body myositis, and used paired single nuclei RNA sequencing and spatial transcriptomics. Again, it was the 10x Visium that we've just heard about, data to profile this tissue. And what they discovered is a selective vulnerability of type 2 myonuclei specific to the inclusion body myositis, and a GUT45A upregulation preferentially seen in type 2A fibers. Luckily, we all got a great introduction already about the different myosin heavy chains and what type of different myofibers we have in this tissue. And what I want to stress out is that they found two different signatures. They found this GUT45A and NORAD signature, which is more genomic stress related, but also a protein degradation marker signature of RNF7 and P62 aggregates, somehow putting labels on this duality of IBM, where we have both the protein dysregulation, but also then the inflammation markers. But I want to, GUT45A upregulation was preferentially seen in type 2A myofibers. And here you can see an overview of the data with a summary of the clustered single nuclei. So we don't look at single cells, but single nuclei, something to keep on top, and spatial transcriptomic data below. Notice that we see the classical massive increase of T cells and inclusion body myositis, but also a decrease of type 2-like macrophages, that are the ones that are classically considered anti-inflammatory. I say type 2 because the nomenclature is also a bit debated, compared to the pro-inflammatory type 1-like macrophages. And now, after describing the cellular composition and transcriptional states of the affected muscle, we wanted to look at the intercellular signaling processes within the muscle microenvironment that may propagate pathology. Because in multicellular organisms, cells don't exist in isolation. In multicellular organisms, such as ourselves, cells receive stimuli from distant cells and surrounding cells, their microenvironment. And now the cell communication describes how cells react to these environmental changes, and which in turn then changes their outgoing signals to other cells, thus building a dynamic network. Local cell-cell communication controls different biological processes, like apoptosis or cell migration, and it classically focuses on protein-mediated interactions, most typically perceived classically as a secreted ligand binding to its corresponding plasma membrane receptor. However, this picture can be broadened to include also extracellular matrix proteins, secreted enzymes, transporters, and interactions that require physical contact between cells, such as the cell or cell extracellular matrix adhesion proteins and gap junctions. Now, we can infer molecular interaction pairs of each biological meaningful group. If I say biological meaningful group, I mean cell types, like type 2 fibers, macrophages, T cells, you name it, and ask the question, how does cell-cell communication differ in inclusion body myositis? Or to phrase it differently, how much does the reaction of cells to stimuli diverge in inclusion body myositis? And to answer this, we used a tool called LIANA, this ligand receptor analysis framework, where the main input that you can see in the middle, which makes it the main part, is our clustered single nuclei data, the biological meaningful groups, and then also databases of known ligand receptor pairs, so we use prior knowledge, it's not unbiased, on the left, and multiple integrated methods on the right. To get a consensus score, because in LIANA we use different methods to somehow concentrate on the overlap of these different methods. And so then we can identify the biological plausible interactions or the ligand receptor pairs in inclusion body myositis. And we usually start with tens of thousands, you can see that on the y-axis here, of interactions that are either upregulated, so on the right, or downregulated on the left. In this case, downregulated means more common in control tissue. Now, the advantage of these methods to generate system-level insights in a hypothesis-free manner becomes one of the major disadvantages, as the authors of single-cell best practices put it quite fittingly, in my opinion. How do we know which ones of these tens of thousands of interactions is now biologically meaningful? And classically, we usually start looking at the top interactions, in this case, the ones hiding at the far right, most upregulated, or far left of the plot. And indeed, we can find some biological plausible interactions at first sight. We can see antigen presentation, T-cell activation from HLA-2 to CD4 T-cells, as we would expect in IBM, which is then antigen-driven response, or first antigen-driven response, rather than just bystander inflammation. And aside from other signaling, we can also see a specific one, in this case, from the beta-2M to KL81, which is specific to the LAMP3 positive dendritic cells towards T-cells. And looking at other cells, at stroma cells, or specifically endothelial cell signaling towards innate immune cells, we can see classical von Willebrand factor to integrin signals. So macrophages are recruited and retained via this interaction. However, I should also mention that we actually also observe a relative loss of this endothelial cell capillary ones, and ones in the middle in inclusion body myositis. And what else can we see in these interactions is that we have Notch and NRP1 signaling from especially endothelial cells from the vascular function towards macrophages, especially towards type 2 macrophages, so the anti-inflammatory ones, which is quite surprising because normally the vascular involvement is not that strong or not reported as strong in inclusion body myositis compared to, for example, dermatomyositis. Whoops, sorry. All these interactions that I've shown you so far are interactions that are more common in certain cells compared to other cells in a single context, inclusion body myositis. However, we are more interested, as I said in the beginning, how does it now differ in inclusion body myositis compared to other groups? And for that, we start classically with differential gene expression analysis, the DEG analysis, comparing the IBM nuclei to those in healthy controls. And you can see here some not so surprising results. We have on the right side, we say interferon or inflammatory signaling like STAT1, CXCL1, PARP1, but we also see a decrease in IBM controlled compared to control tissue of like certain kinds of integrins or also other tissue remodeling factors. And now, if we input those into the Liana platform again, we can see now that the interaction strength on the right is more towards the Gcom red, the one on top here, if the receptor and the ligand are both upregulated in IBM compared to control tissue, or more down if they are more active in healthy controls. And what we can see is that in inclusion body myositis, we have stromal and muscle resident cells that actively orchestrate immune activation and tissue remodeling through, for example, complement interactions. Here, so we have complemented media danger signal propagation from especially these fibroid adipogenic progenitor cells towards type 1 macrophages. And now that I want to mention that when we look at differentially expressed genes, we also lose some signals. So it's always like some kind of scale that we want to look this at, because we also lose immune interactions because we simply don't have that many dendritic cells or like classical T killer cells in healthy tissue. Luckily, we don't have that. But that also means we can't really compare the signal between inclusion body myositis and healthy tissue in that case. So we lose some part of the tissue. Yet, after looking at the upstream signals that start the reactions, the ligands and the receptors, we also looked at the other side, so the more downstream propagated signals. And we did this via a tool called NicheNet, which tells us how likely a ligand of interest is the cause of observed downstream changes. Here, the ligands are strongly upregulated in their actions towards inclusion body myositis myofibers, and we can see that they are also able to explain part of the downstream response in those, with interferon gamma explaining it best compared to other statistically plausible ligands. And interestingly, interferon gamma also belongs to a subset of top ligands that is specific to damaged but not healthy nuclei. And to better understand the NicheNet score, you can also see here what it actually does is that we look at downstream targets that are inferred, and then we look at which ones are actually dysregulated. So for example, STAT1, TAP1, STAT2, TAP2, and so on. And to finally infer the functional relevance, we use the third, and for the purpose of my talk, today's final method called RIDDEN, which then takes expression data and based on its perturbation studies, outputs how likely certain receptors are activated. And as you can see, it also agrees with the NicheNet scores that interferon gamma targets like interferon gamma receptor 1, interferon gamma receptor 2 are more up in the middle here in inclusion body myositis compared to other compares. And finally, to test for spatial relevance of inferred interactions, we can go into the Visium data and then look at certain interactions that are maybe spatially less relevant or spatially less interesting to us, which are more propagated throughout the whole slide like this complement interaction here on this IBM data. But we also see spatially meaningful interactions, so some that are enriched more to the red. That means that both ligand and receptor are expressed more on the spot, or depleted, so that one is up and the other is down, or the other way around. So, and with that, I want to conclude that on the biological and medical side, the tools that we employed did reveal coordinated antigen presentation and extracellular matrix-mediated immune regulation within a chronic inflammatory niche. And we argue that this depicted influence of non-immune cells does play a role in inclusion body myositis' infamous therapy resistance. I want to thank many people, first and foremost my own group and home in Germany, my advisors, our collaborators, but this is only a small subset of all the people, and one slide can only hold so much space and also time, I know. So with this, I just want to say thank you, obrigado.
Thank you very much. Beautiful talk and kept to time. Please make yourself known to us if you have a question while people are just thinking. I will just share a thought, which is it's extraordinary how many immune mechanisms actually are clearly shown by your data in IBM. And so do you think we can really distinguish the different pathological mechanisms between the types of IIM with these kind of data? If you were to compare IBM with another of the IIM diseases.
Sorry, I didn't understand the beginning of the question.
So I guess my question is, how well do you think these in-depth pathway analyses, which you've used, would be able to distinguish specific mechanisms between the different clinical phenotypes of the myopathies?
Mm-hmm. So we only started with this analysis, right? So we looked at more broad data. We didn't actually assemble a cohort specifically designed to address this question or to use these tools. So I think for now, it would be very difficult to do so, honestly, frankly. But I think if we actually try to design our cohorts more clinically relevant and targeted towards this question, I think that will definitely be possible in the future. Yeah. I think the field is progressing incredibly fast. There are more and more tools. Also, I'm very happy about that, more and more accessible to the wider public, and I think that we will definitely be able to see that. We just have to use the tools now in the right way.
Yeah, I absolutely agree. I think we do have to move on because we have to be on time for the opening ceremony. So I'm going to pass back to my co-chair for last but not least.
So let's keep with our excellent timings. So I now call you Vi Lu. And she's speaking about the plasma. Oh, the title that I have here is slightly different, but we'll follow your title.
Yes, I can repeat.
Which for sure is the right one.
Yes. Hi, everyone. And today I will present our study using the Olink technique to profile myositis plasma and find tissue-specific reactions. Let me see, how can I move this? Yes, I have nothing to declare. So we know that the bloodstream is a dynamic communication system that carries not only cells, but also a wide range of proteins and molecules coming from different tissues. And these molecules enter circulation either passively when the cells die and release the contents, or actively through secretion, such as cytokines and exosomes and hormones. And in myositis, the blood biomarkers that we routinely use in the clinic only reflect a small fraction of a complex disease. What we're lacking here is a broader proteome level view of what is happening in myositis circulation. So our study is part of a larger collaboration between Karolinska and our national infrastructure SciLife lab. So this collaboration used the Olink technique to profile 1,400 blood proteins in the plasma samples from 6,000 patients across 59 major diseases, with the initiatives to establish a pan-disease blood atlas. Building on that work, we further analyzed myositis plasma coming from the Swedish National Myositis Registry, SWIMARNet, with the aim to identify specific protein signatures in myositis circulation. And in total, we included 200 patients. I also collected their lab test results taken 3 months within sampling time, and disease activity measurements 6 months within sampling time. And this is the composition of our patient cohort. So due to time limitation, I'll be focusing on antisynthetase, dermatomyositis, and immune-mediated necrotizing myopathy today. So we begin our analysis by differential abundance analysis by comparing one myositis subtype with the rest of the subtypes. So dermatomyositis showed 62 enriched blood proteins. And in these blood proteins, we first see interferon-related proteins, and we also begin to see tissue proteins. For example, the top-enriched protein, keratin 5, together with keratin 14, are expressed in both lung and skin. And we also see quite a few liver-associated proteins and matrix-associated proteins. And when we were to compare the two most common dermatomyositis-specific autoantibody group, you see the remarkable difference between anti-MDA5 and anti-T1 gamma. So anti-T1 5 looks very much different from the rest of the autoantibody groups with 222 enriched proteins. And again, we see interferon-related proteins and lung-associated and liver-associated proteins. And on the contrary, anti-T1 gamma only have one significantly increased protein, which is this lung and skin protein, keratin 5, suggesting that the proteome of T1 gamma is not much different from the more general myositis population. And then as 80% of our anti-Senescence patients are positive for anti-Jo1 antibodies, so we can focus on this antibody group. In the enriched proteins, we again see quite a few lung-associated proteins, and the top enriched protein is this surfactant protein D. And then we also have skin-associated protein and tissue response proteins. And for Jo1, it seems that there is a more specific type 2 interferon, type 2 interferon enrichment. And anti-HMGCR antibody accounts for 90% of our IMNM patients, so the differential abundance results looks pretty similar between the subtype group and the autoantibody group. And in the enriched protein, we have quite a few muscle proteins. And in these, we have MEGF10 and NCAM1, which are involved in muscle regeneration. And we also have extracellular matrix and fibroblast-associated proteins. And we also know that extracellular matrix remodeling is also involved in muscle regeneration. So it seems that in the blood proteome of IMNM, you don't only find proteins that indicate muscle necrosis, you also find signs of muscle regeneration. And so that we have identified tissue proteins enriched in different myositis subtypes, and these tissue proteins seem to correlate with the preferentially involved organs in these subtypes. But would they be correlated with clinical symptoms and lab results regardless of the subtype? For that, we did a correlation analysis. And indeed, the proteins are strongly correlated with the levels of creatine kinase or other muscle proteins. And for the CK-correlated proteins that are not muscle proteins, most of them are extracellular matrix associated. And then we also have this interleukin 32, which is a pro-inflammatory cytokine that promotes the production of TNF-alpha. It could mean that IL-32 is more potent in inducing muscle damage, maybe. And as a comparator, the proteins that's correlated with levels of CRP are either other pro-inflammatory molecules, such as interleukin-6, or more ubiquitously expressed proteins, suggesting that CRP has a more pleiotropic effect. And the proteins correlated with interstitial lung disease are many other lung-associated proteins, such as keratins and surfactant proteins. And we also find that the top enriched and lung-associated proteins, they have quite a good discriminative performance to differentiate IOD from non-IOD. And we also find that if we were to combine the top enriched proteins, their discriminative performance is even slightly better than individual ones. And for the sake of time, I will skip the skin part. And we also compared IOD patients with either MDA5 antibody or with Jo1 antibody. What we find is that MDA5 IOD is actually enriched in all three types of interferon pathway-related proteins, and as well as liver proteins. What's more interesting is that extracellular matrix and surfactant proteins are enriched in both MDA5 and Jo1 IOD, but with different specific members. So for example, the matrix protein that's enriched in MDA5 IOD are mainly matrix scaffold proteins and involved in fibrosis. While the Jo1 enriched matrix protein, they mainly work for matrix degradation and cell migration and basically matrix remodeling. And the MDF5 enriched surfactant protein is surfactant protein A, while surfactant protein D is enriched in Jo1 ILD. And so we found tissue proteins enriched in different myositis subtypes, and they seem to correlate with different clinical symptoms and lab results. But these are more on a single protein level. What we want to approach the question whether there is a tissue-specific response in myositis plasma in a more collective way. For this, we did a tissue enrichment analysis by calculating a tissue score. We're hoping that this tissue score would represent the degree of tissue damage. And indeed, the tissue scores are different in different subtypes. For example, IMNM and HMGCR patients showed the highest skeletal muscle score, suggesting that they have the strongest muscle damage. And anesthetist patients have the highest lung score. But in terms of autoantibody groups, it is the MDF5 patients that have the highest lung score. And dermatomyositis, in particular, MDF5 again showed the highest liver score. To confirm the elevated liver score really means that muscle damage, then we went back and reviewed the liver function tests of our MDF5 patients that's been done in the hospital. We chose TIF1 gamma as a comparator group. So at first glance, the levels of ALT and AST are not different between the two groups. But then we know that ALT and AST, they are both expressed in liver and in skin. So if we're to divide them by the levels of CK as a way to adjust for muscle damage, and then we see that the ratio is actually higher in MDF5 than in TIF1 gamma. And if we're to look at more liver-specific biomarkers such as alkaline phosphatase or gamma-glutamyl transferase, and their levels are indeed, again, higher in MDF5 than in TIF1 gamma. However, we didn't find any remarkable imaging or pathological changes in our MDF5 patients. So we conclude that the elevated liver score in MDF5 does indicate liver damage in this group, but this damage is more subclinical. And to conclude, our study has identified proteins enriched in different myositis subtypes and correlated with clinical symptoms. And some of these enriched proteins may have the potential to serve as biomarkers for tissue damage. And we have identified that different biological processes activated in MDF5 ILD and Jo1 ILD. And our MDF5 patients have the strongest liver and lung damage as represented by the elevated liver and lung tissue score. And finally, the blood proteome of patients does not only show the systemic changes, it could also reveal distinct tissue-specific involvement and this involvement would correspond to tissue damage and disease activity. So as acknowledgement, I thank my collaborators in SciLife Lab for performing the Olink experiment, and I also thank the whole KI myositis research group, in particular our good physicians and nurses. It's their dedicated work on myositis 3 MyoNet for decades that enables this project to happen, and of course all patients. And I also thank Shanghai Hospital for my postdoc scholarship. Thank you.
Thank you. This was an amazing talk and amazing results. We have time for 1 to 2 questions. Okay.
Question here.
Okay. Please.
Do you need a microphone? Could you bring a microphone to this? Yeah, it's coming. Sorry.
We can start by this question and then move to the other one.
Yeah, very quick questions.
Okay. Very quick question. 2 questions. Are all patients treatment naive or treated?
Oh, yeah, that's a good question. So by the time we did our analysis, we didn't have enough treatment information for these patients. We have treatment information for about 40 patients, and we compared the treatment naive and the treated patients, and we didn't find any differentially expressed proteins between the 2 groups.
I think there's a second quick question.
This main question is, you mentioned that the anti-synthetase ILD and MDR5 ILD had both upregulation of ECM proteins. But are there any biological meanings?
Yes, I think there should be. As I said, I think the matrix protein that's enriched in MDR5 ILD, I would say it's more malicious. It works for this irreversible fibrosis, while for Jo1 ILD, you still have this matrix remodeling. So it's kind of like a repairing system process for Jo1 ILD. So I think it's slightly more benign, I would think, compared with MDR5 ILD. Yes, thank you.
Thank you.
Really was just kind of about the similar part to, or similar question to his first one. But to be even maybe a little more expansive, have you looked at any specific of the treatments over time to see if you see any changes that may correlate with the clinical response or a non-response?
Yes, I think that's a very good idea about our future plan. And this is more of a cross-sectional study, so we didn't include samples at different time points, but I think in the future it would be a good idea to compare.
It's great. We'd love to talk about it with you later.
Treatment, yes, thank you. Thank you.
Okay, so we're going to close, and I believe that the opening session is immediately after.
So I understand the opening session is coming literally in 2 minutes, so don't go far away. I guess a couple of people might need a very brief break, but be back because you have a very exciting session about to begin. See you again. After all that time. It was a good session, actually. Bizarre to put this first. On here, I kept a few notes. Only my notes. The rest.
Please keep it.
You sure? Just. Oh no need. Thank you. I know you like it. Thank you. Work out how many people are yeah, I think, and then we... Tá bem. Okay.
We have some. Oh, you have some. I brought mine.
Certeza absolută.
Extra one? Yeah, the extra one is coming. So,
you're doing the story. I don't know.
I didn't want to be too specific, not more around. It's kind of recent on me, so...
Vão ser as 3 ou mais oradores?
Não, não, somos só os 3.
I think Raquel asked.
Yeah, it was not a...
There are a lot of patients involved.
So you were all...
Yeah. Maybe someone gets up on the side.
Maybe you can
Or should someone go outside and push everybody? Stine can do that.
Onde eu tive sessão dos pacientes lá em cima e foi a horas atrasou. Aqui também, eles acabaram mesmo mais 45. Estivemos a fazer yoga facial, foi muito engraçado. Yoga facial. A Maria Isabel, Sou Maria João, tem que ser Maria. Não, Maria João. A Maria Isabel é que familiar do Joaquim. Eu sou amiga da Débora, a mulher do Joaquim. Eu tinha uma avó, mas coincidiu com a minha ausência, então depois chegou para a doutora Sofia. Muito bem. Eu gosto muito da doutora Sofia. As coisas na vida nunca acontecem por acaso. Quando fui diagnosticada, felizmente conhecia o Joaquim.
Well, it's quite a long session, I think.
Yeah.
From 1 o'clock.
Yeah, and people get stressed of being
Yeah.
quiet for a long time.
And then they want to stretch their legs.
Yeah. We would just ask to open some space for the patients to get closer. But shall I ask? Shall I start?
Yeah. I think it's a good idea. We said it.
May I ask your attention? Good afternoon. So, good afternoon, everyone. My name is Raquel, and I'm honored to share this Congress. On behalf of the organizing committee, it is my great pleasure to welcome you in Lisbon for the 6th Global Conference on Myositis. This meeting, held every 2 years, represents a key moment for the global myositis community. The year 2026 stands out as a particularly exciting time, as we are witnessing significant advances in the understanding of disease pathogenesis, the identification of biomarkers, and the development of innovative treatment strategies, all with an increasing focus on patient-centered care. Over the next few days, we bring together clinicians, researchers, patients, industry partners, and advocacy groups, all united by a common goal: to improve outcomes and quality of life for people living with myositis. I would like to begin by acknowledging all those who made this Congress possible. First, our local organizing committee, who over the past 2 years have dedicated countless hours to meetings, planning, and coordination, working diligently behind the scenes to bring this Congress together. Their dedication, generosity, and rigorous attention to detail have been truly remarkable. Thank you for that. Second, the steering committee of this Congress, with whom we have met monthly, and you can see from their faces, not every time we were so happy, as you can see it from the Zoom, unfortunately, but that's life, with whom we have met monthly over the past 2 years, and whose guidance and support have been fundamental in shaping and supporting this meeting. Thank you all as well. I would also like to recognize our International Scientific Committee, composed of leading experts in the field, whose guidance ensured scientific excellence, relevance, and balance across clinical, translational, and basic research. My thank you for them as well. I'm extremely happy because I can see that this Congress continues to grow, has a truly global platform. We are delighted to welcome participants from a wide range of countries, reflecting the international nature and collaborative spirit of our community. This year, we had almost 680 participants, which is an amazing number. And we can, as you can see from the map, they came across from all over the countries and from several continents. So I really, really appreciate the interest that has been growing in the myositis field, and thank you for that as well. We also received a record number of abstract submissions this year of excellent quality, which made the selection process even more challenging. For this reason, I would like to once again thank the members of the Scientific Committee for their careful review and dedication. We have received almost 400 abstracts this year that will be presented either as oral presentation or as poster in the format of poster tour, poster hanging presentation, or online poster presentation. And I also would like to highlight that all accepted abstracts will be published in a special issue of Clinical and Experimental Rheumatology, which, as you know, is the official IMEOS journal, ensuring dissemination and visibility for the work and efforts of all the contributors. Congratulations for that as well. Over the next 4 days, we have an exciting and comprehensive program ahead of us. You will experience a rich and dynamic scientific program, including state-of-the-art lectures, translational science updates, clinical debates and controversial sessions, oral and poster presentations, interactive workshops and networking sessions, and sponsored symposiums. We also highlight the importance of fostering the next generation of researchers with dedicated sessions aimed at early career investigators, providing opportunities for discussion, collaboration, and growth. And we are also very proud to announce that the program has been awarded 23 credits by the European Accreditation Council for Continuing Medical Education. So thank you to all that contribute for the scientific program. We had started with a great muscle ultrasound course this morning, and this is just some pictures from the course, which included world experts in ultrasound, several patients who generously volunteered as models, and 38 participants who actively engage in hands-on muscle ultrasound practice. I apologize for the ones that unfortunately could not make it. They were on the waiting list, but for sure, more courses of this will be disseminated and happening all over the world. Another key priority for us is close collaboration with patients. Our collaboration with patients and patient organizations continues to grow and remains fundamental to everything we do. Listening to patients is therefore of utmost importance. For this reason, we believed that patients should be involved throughout the program. In that spirit, most scientific sessions will begin with a patient sharing their perspective. And in parallel, there will also be multiple sessions and workshops led by patients themselves. And thank you, Karen, for sharing the slides with me. The Congress app is now available and can be downloaded by following the steps shown on the screen. Please, I suggest you to download the app for real-time updates and interactive sessions. You can also follow us on our social media channels, where news and daily highlights from the Congress will be shared throughout the meeting. We would also like to acknowledge and thank the European Accreditation Council for continuing medical education, the IMEUS, the International Myositis Society, the Portuguese Society of Rheumatology, and the Portuguese Society of Pediatric Rheumatology for their scientific support and endorsement. We also acknowledge our sponsors, whose contributions make this event possible and play an important role in advancing research and therapeutic innovation in myositis. We also would like to thank MIRA and MCTC for awarding several travel grants, which have enabled many colleagues to join us at this meeting. Again, we also would like to acknowledge Myositis UK, that has once again opened the opportunity for research grant awards. This year we received 20 candidates, of which 6 outstanding projects have been selected and will be presented here at Gcom during the speed funding session on Wednesday at 8:30. The best 3 projects will receive a grant of 18,000 euros each, and the winners will be announced during the closing session. So please stay tuned till the end of the conference when the results will be announced. Today, and please don't leave it, today at 7:15 we will have our welcome reception, which will take place in Auditorium 6 and 7. You are all warmly invited to join us, and at the end we will have a small surprise for you, so please stay tuned. On the 25th, we will have our social networking event, which will take place at the emblematic Pátio da Galé. This beautiful venue, known for its impressive halls, regularly hosts some of the most important national events. Pátio da Galé is located in Terreiro do Paço, one of the most iconic and attractive places in Lisbon. So, if you enjoy good food, great company, and a bit of fun, I warmly invite you to join us for the evening. There are still a few places available, but registration is required, and you can do that on the Congress website. And of course, a few surprises are waiting for you. On March 24, just to let you know, please don't panic, between 10:30 and 12, there will be a citywide tsunami alert system test in Lisbon. And as part of this exercise, sirens may be heard in several parts of the city, and it's possible that they will also be heard here nearby. So please do not be alarmed, this is only a scheduled test. I repeat, this is only a scheduled test. And finally, our sincere thanks to Steen and Stefano on behalf of EAM Services for their incredible support and commitment from the very beginning of this journey. Always bring together such positive energy along the way, always with a smile. Finally, I would like to wish you all a wonderful Congress, and thank you for being here with us, both in person and virtually. Thank you. It is now my great pleasure to introduce our next speaker, Maria João Mendes, a patient representative from Portugal, who will share with us a little of her story. Thank you. The floor is yours.
Good afternoon. My name is Maria, and it's a real honor to welcome you to Lisbon and to this year's Gcom. My journey with myositis began in 2021, shortly after recovering from COVID. It seems my immune system decided that surviving a pandemic wasn't dramatic enough and chose to add its own creative twist, dermatomyositis, complete with bold, expressive, and much uninvited skin symptoms. For me, those skin manifestations quickly took center stage. They appeared in colors and patterns nobody asked for, often behaving like surprise special effects. I quickly learned that SPF is no longer a simple product, it is a lifelong commitment. At this point, sunscreen and I have what feels like a legally binding relationship. Humor aside, dermatomyositis has required adjustment. It has meant listening carefully to my body, navigating uncertainty, and reshaping daily routines around what my skin and sometimes my muscles decide to do. It has taught me patience, resilience, humility, and the value of advocating for myself in medical spaces that don't always expect a rare disease to walk through the door. Though I was used to get weird looks when I say I have celiac disease, I got another autoimmune on my bingo cards. Luckily and thankfully to my physician's team, everything is under control now, and most of the days I don't even remember I have this condition. Before my diagnosis, I didn't know anything about myositis, and frankly, I didn't know anyone else living with it. My experience with the broader myositis community is still growing. I believe small moments, like conversations and shared stories, are quite important. A shared understanding and the comfort of knowing that this path, while rare, is not walk alone, brings some kind of solace. I look forward to learning more from all of you in the days ahead. And this is precisely why this Congress matters. To the organizers, thank you for the extraordinary dedication it takes to bring an international event like this together. The coordination, the communication, the planning, none of it is simple, and yet you have created a space where science, collaboration, and humanity truly meet. We are all here because of your vision and hard work. To the researchers and clinicians and the industry partners, your commitment is the backbone of our hope. Every study, every paper, every clinical insight you bring into the world has the potential to make our lives better, whether through clear diagnosis, improved treatments, or simply a deeper understanding of what we are facing. Your work goes far beyond labs and clinics. It reaches right into our homes, our routines, our futures. For that, we are deeply grateful. To the patients and the caregivers among us, your resilience is profound. Whether you are newly diagnosed or have lived with myositis for many years, your presence here is a testament to strength in all its forms. Thank you for showing up, for speaking up, and for helping shape the future of care. Não sei o que o futuro trará. I know not what tomorrow will bring. From the Portuguese author, Fernando Pessoa, always has been one of my favorite quotes. For those of us living with dermatomyositis, that line resonates strongly. We often don't know what tomorrow will bring, whether it will be a calm day or a flare, a day of energy or a day of rest. But what we gain from gatherings like this is something powerful, the reassurance that tomorrow may also bring progress, learning, and innovation, and that none of us face uncertainty alone. I'm looking forward to all the patient voices in the scientific sessions and the patient-led workshops. Please join us. These workshops are open to everyone. Thank you for all being here. I wish you an inspiring, meaningful, and hopeful Congress.
Thank you so much, Maria. It was great to hear you. It is now my pleasure to introduce Anneke van der Kooi, the IMEOS chair. Anneke, the floor is yours.
Thank you very much. I'm just, giving a very short presentation. Tomorrow, at the end of the day, there will be the IMEOS General Assembly for the members. And, well, IMEOS has been established. Maybe the next slide can come. I don't see it yet. It has been established in 2000. I should do it myself. Sorry. Thank you very much. IMEOS was established in 2019, thanks to Jens Schmidt, who was sitting over there, who took the initiative. So thanks for that. And well, the IMEOS was established to provide a global infrastructure with the aim to harmonize standards of care, diagnostic criteria, classifications systems, and was also to promote collaboration between expert and patient groups. But one of the main topics is also the education. Sorry. And the first meeting was in 2015. At that time, it was not an IMIOS meeting yet because the IMIOS did not exist. But the first international meeting on myositis was organized by Ingrid Lundberg in Stockholm. And at that time, we had 150 participants. Later on, almost every 2 years, except for the COVID problems, I cannot see it when it's stand here. The meetings were in Potomac, in 2019 in Berlin, in 2022 in Prague, 2 years ago in Pittsburgh, and now in Lisbon. And it's the 6th, you already said it, the 6th Gcom meeting. And at this time we have more than 600 participants, so an enormous growth of the Gcom organization. As I said, one of the primary missions is the education of myositis specialists. And well, now, a long time on the side was the save the date, but now is the date that the Gcom started. We're all here, an awful lot of people, and I hope that we have a very nice meeting. And I thank the local organizing committee, a special recall, Eduardo, Anna, Sofia, and Jor. Thank you very much for all the work that you put into this conference. I know it has been an awful lot of work, and we hope we have a very nice conference. And I wish you all a very good conference. So thank you.
Thank you, Anek, for your great and really nice words. We are now closing the opening session. I invite you to join the coffee break. After that, we'll have the posters and the poster tours, and then the symposium. And please don't forget 7:15 here for a surprise for you. See you later. Thank you.
Okay, good afternoon. So we want to thank the organizers for giving us the opportunity to present more details and more information about CAR-T for myositis. And I will leave this for a few minutes so you can read the entire disclaimer. This is required by our legal team. And these are our disclosures. And we are honored to have Professor Chenoy here, who will be presenting on some additional information after my brief introductory remarks. Ooh, we're off here. Okay, so in terms of our learning objectives, we'll cover a number of items. So one, we'll review the key lessons of CAR-T therapy from hematology and in autoimmune diseases. Hold on a second. Just 1 second here. Oop, I messed that up. Okay. I want to be able to see my slides. But so the second is that we'll describe the rationale for deep B-cell depletion, particularly by targeting CD19 in the treatment of myositis. And third, Dr. Chenoy will present and describe the late-stage clinical study designs and review the latest results from the RESET myositis study. Oh, hold on a second. Yeah. Okay, here we go. So thank you for your patience. So I'm going to give a quick overview of CAR-T and autoimmune diseases. So we've talked a lot about CAR-T therapy already, and Professor Boyer has already discussed quite a bit about B-cell involvement in myositis and what's the rationale and justification for that. So I'll just give a very brief overview of CAR-T, and you can see the diagram on the left-hand side, which I'll go into a little bit further later on. But what are CAR-T therapy? Is chimeric antigen receptor T cells. The reason it's chimeric is actually because it's a synthesis of multiple different domains. It isn't a natural occurring molecule. It's a synthesized product. And you can see here that there's an antibody fragment, there's a co-stimulatory domain, there's a signaling domain, there's a transmembrane domain, there's also even a hinge region. So it's actually a chimeric in the sense that you put together a synthetic molecule that does not exist normally. So there have been 7 CAR-T therapies that have been approved, of which 5 of them are targeting CD19, which are found on largely B cells. And 2 of them are targeting B-cell maturation antigen, which is found typically on plasma cells. And the way CAR-T cell therapy works is that they bind to their target antigen, CD19 or BCMA, and lead to the killing of that molecule through the release of various cytokines. So there's been over 35,000 patients in the hematology oncology world who have been treated with a CAR-T therapy. So there's a lot of lessons that we can learn from that. First of all, we know the efficacy is quite good. And these are patients who had B-cell lymphoma, B-cell leukemia, who were refractory to multiple different rounds of chemotherapy and still had severe disease. And you can see that the complete remission rate was achieving up to 67%, and the long-term remission rate was also up to 60%. The second thing that we've learned from these over 35,000 patients is that we know what the safety is. So there's cytokine release, and there's neurotoxicity called ICANS. We know about B-cell aplasia. We know about infections, insertional oncogenesis as a potential risk, as well as secondary malignancies. So oncology has already informed us what we need to know about CD19 CAR-T therapy. So being very familiar with this, in fact, many different oncology centers are now treating patients as outpatients because they know what to expect and they know how to treat these potential side effects. But what we also know is that the adverse events with CD19 CAR-T therapy may be reduced in patients with autoimmunity. We've learned this from Professor Schett's patient cohort that he's treated in Germany. And we've also heard about this from various other patients that have been treated in clinical trials. And the reason we know this is that we can look at the various risk factors that we have in oncology that do not exist in autoimmunity. For example, in oncology, there's a very high B-cell burden. A lot of B cells, malignant B cells, and therefore they lead to having higher rates of CRS and neurotoxicity, as well as more severity. And in oncology, they tend to see longer persistence of CAR T cells, which can lead to prolonged B-cell aplasia, which in turn can lead to increased risk of infection. These we don't see in autoimmunity. You don't see high target cell burden, you don't tend to see CAR T persistence, and you don't have prolonged B-cell aplasia. And of course, in oncology, there's mutational load. So this mutational load can lead to secondary malignancies, or it can lead to potential relapse due to antigen escape. And we don't have that in autoimmunity. So 4 of the key risk factors in oncology for safety events are not present in most patients with autoimmune diseases. So we've seen lower rates of CRS, lower rates of neurotoxicity, lower rates of infection, et cetera. So we have a product called Rese-cel, and it's a fully human CD19 binding domain. You can see that in the maroon area in the middle, and it has a 4-1BB co-stimulatory domain. So the first thing to know is that because it's fully human, you heard earlier today about a question about immunogenicity. Because it's fully human, there's a lower risk for immunogenicity. There's no chimeric, it's not a chimeric in the sense that there's no mouse particles or mouse fragments in the binding domain. And second is because it has a 4-1BB co-stimulatory domain that tends to confer a lower risk of cytokine release syndrome and neurotoxicity. So how do we make CAR T cells? So in this particular setting, at least what we have today, is autologous. So it's these T cells are derived from the patient's own body. And it's derived and collected through a process through leukapheresis. The T cells are isolated, and these T cells are reprogrammed in the laboratory setting using a viral vector to deliver the gene that encodes the CAR. And then it's proliferated in the bioreactor so that you have a lot more CAR T cells which are activated. And then you have to prepare the patients, maybe, and maybe not, right? So right now, what oncology has done for every patient is they give patients pre-treatment with fludarabine and cyclophosphamide, which are chemotherapy agents that are used for treatment of B-cell leukemia and lymphoma. And there's various reasons for that. Namely, you want to reduce the tumor bulk, you want to potentially get rid of the competition so your CAR Ts can be very successful. But it's possible in the autoimmune setting that preconditioning may not be needed, and that's being explored. And finally, the CAR T is infused via IV infusion. So I just want to give you, as my final slide, an update on where we are with a clinical program for Rese-cel, which is a CD19-directed CAR T therapy. It is a disease, so we're looking at various disease-specific cohorts you can see listed here. We're looking at 5 different indications at myositis, and you can see the 3 that we're looking at. So you can see juvenile myositis is included. We're looking at lupus, scleroderma, myasthenia gravis, and pemphigus vulgaris, which is where we're looking at no preconditioning options so that the patients don't have to get cyclophosphamide or fludarabine. And I did forget to mention at the very beginning that there are QR codes. So if you're taking photos, we can show you the QR code again. You can get the whole slide deck in a very nice format, so it'll be nice to in your photograph. And if you have any questions later on, there'll be a QR code that'll come up again that you can punch in the questions that you want to ask. So with that, I'm going to turn it over to Professor Chenoy.
Thank you, David. So I don't need to present this slide to the audience here. We know that there's a big unmet need in patients with myositis, with a high disease burden in terms of disability and mortality, and emerging risk of things like cardiovascular disease as well, which has been reproduced in multiple studies globally. And we all know about the massive steroid burden that these patients have, and it's just the sort of population where you don't want to be giving steroids. Generally a female population over the age of, average age over the age of 50, and many of these patients remain on steroids years after their diagnosis. I thought this slide was interesting because it's trying to quantify the actual numbers of patients that we're dealing with, and it's approximately the same in the US that it is in Europe as well. So we're talking roughly about 80,000 patients in both areas. And if we split that down, we're talking about 60,000 patients with dermatomyositis, about 10,000 patients with antisynthetase, and about 16K with IMNM. And you can see here on the right, the UK's officially left the EU, so it's EU plus UK. And again, I don't need to go into this slide in detail, but we're now classifying myositis more according to the antibody status and clinical features rather than a PMDM classification, which allows us to work out the underlying immune mechanisms for each of these subgroups, the clinical characteristics, and then what is the tailored treatment that we need for these particular clinical subgroups, be it DM or antisynthetase or IMNM. But what we do know is that B cells do play a central role in these conditions, including other autoimmune diseases as well. But what we're using, the current paradigm, rituximab, they often result in incomplete B cell depletion in the target tissues and the lymphoid organs. On the left, you can see a number of strategies for targeting the B cells, and you'll be most familiar with CD20 and CD19 inhibition. And there's multifaceted autoreactivity that extends not just beyond the bloodstream, where we're measuring things like CD19 levels, but into the target organ tissues as well. So the failure to achieve remission in the patients that we're giving B-cell therapy to at the moment may be the result of incomplete B-cell depletion. Let's just remind ourselves of which CD markers are present on which B-cell subsets. And you'll be familiar with CD20, with rituximab, which is going from the pro-B cells to the memory B cells, but not targeting the more mature B cells, the plasmablasts and the plasma cells. And CD19 has a slightly wider repertoire. But if we're wanting to target the more mature populations, then it has to be CD38 BCMA. So on the right-hand side, we can see the lymph node biopsies from patients who have respectively received obinutuzumab, blinatumomab, rituximab, and CAR-T. And you can see the degree of B-cell depletion in the CAR-T patient compared to the patients who've received these other therapies. And then again, the middle column is CD19 expression, and the bottom row is CD20. So these aren't specifically IIM patients. This is a range of patients who have received these drugs. So small study, but suggestion that the CAR-T is achieving a deeper B-cell depletion compared to the other B-cell-based therapies, and including bispecific T-cell engager therapies. But more work needs to be done to look at this in more detail, and also to define what this immune reset really means in these patients. You all know about the TIS and the IMAX core set measures, but what you may not be aware of is the emphasis that's placed on each of the core set measures. So this is a really neat slide that shows that there's more emphasis on the MMT and then extramuscular activity and physician global activity compared to the other three measures. And this can be an issue when you're dealing with clinical subtypes. So for example, in DM, there may be more of an emphasis on extramuscular disease activity compared to muscle involvement. In antisynthetase syndrome, there may be more extramuscular involvement. And IMNM, the MMT8 may not accurately represent what's actually going on with patients with very proximal involvement, and the extramuscular score becomes largely redundant in IMNM patients as well. So we just have to bear these issues in mind for the different subsets. So building on what David's already presented, the RESET myositis, which is a phase 1/2 open label study enrolling IAM patients with moderate to severe disease, refractory to standard of care, and the primary endpoint was the incidence and severity of adverse events at day 29. So the patients received leukapheresis, and the rese-cel allows the production of the CAR T cells. And just prior to preconditioning with fludarabine and cyclophosphamide, the patients discontinuation of all immunosuppressants. And then they have a single infusion of rese-cel, and then the primary endpoints and the secondary endpoints. So patients had to have either DM or antisynthetase or IMNM, and based on the presence of MSAs, and juveniles were also allowed entry as well, aged between 6 and 17. So these are the baseline characteristics of the first 13 patients, so we'll just walk you through this. We can see the mean age, first of all, in the first row, and then the proportion of females, and you can see that there's an overrepresentation of male patients in the IMNM patients. The years since diagnosis is variable, but we have to bear in mind the low patient numbers. So, for example, in the ASAS cohort, there's only 2 patients in there. All of the patients had an MSA. The DM, it was TIF1 gamma, NXP, and SAE. Both of the ASAS patients had JO1. There was 1 HMGCR, 1 SRP, and 1 NXP2 in the JDM patient. The mean MMT8, what's really interesting is it was actually lowest in the DM group, and the CK was within normal limits for the DM group as well, and as you'd expect, higher in the IMNM group. Most of the patients, the majority of patients had received prior rituximab, IVIG, and at screening, the majority were on systemic glucocorticoids and at least 2 immunomodulatory treatments. So if we look at the SAEs, and this is looking at CRS and ICANS, 4 of the patients had grade 1 CRS, and none of the patients had any episodes of ICANS. There was no serious infections recorded. And related SAEs, there was 1 episode of febrile neutropenia in the juvenile patient. If we look at on the left-hand side is the pharmacokinetics of the drug, and we can see an expansion of rese-cel as one would expect following the infusion, followed by a marked reduction after about 30 days. And on the right-hand side, we can see the complete but transient peripheral B-cell depletion occurring within 1 to 2 weeks and persisting till about 30 days post-infusion. And the peripheral B cells began repopulating 2 to 3 months afterwards with transitional naive cells, indicating the potential for a B cell reset. So let's look at the DM data in detail. Of the 4 patients who received rese-cel, all of them discontinued their immunomodulatory treatment, and they were free of treatment at week 16, either on low dose or no glucocorticoids, and 3 of them had a major TIS response recorded. The 4th patient, there's not enough data to know what's happened yet. And if we look at a separate endpoint of the pivotal study, these patients are achieving a moderate TIS off all immunomodulatory treatment on no steroids or low dose. For 2 of the patients, they were able to record a reduction in antibody titer as well. So if we look at the CK, it was pretty low at presentation, so we can't tell very much from that data. On the right-hand side, we can see the MMT8, and for the 3 patients that were followed up, to week 16, you can see a marked improvement. And if we look at the SIDASI score, we can see a significant reduction in 2 of the patients. In one of them, the SIDASI level was pretty low anyway, and we can see some pictorial representations of the improvement in the rash. So next, if we look at the 2 ASAS patients, and the response is somewhat different from the DM patients. We can see the first patient did achieve a major response, and this went into week 20, but then after week 24, there did appear to be a relapse. And the second patient did not achieve the major TIS response. And in that particular patient, there wasn't a reduction in the antibody titer. If we look in more detail, we can see a transient increase in one of the CKs, which then goes back to normal. And then for the MMT8, there did appear to be an improvement in the MMT8 scores in both patients, yet the patients did not achieve the necessary endpoints. And then the IMNM patients, there were 6 of these patients, and 3 of them achieved the prerequisite response, but not heading into the major category of TIS. So yet 3 of them did manage to discontinue all immunomodulators and were on low dose or no glucocorticoids. And a complete transient B-cell depletion was noted in all of the patients that we've talked about today. So if we look in more detail, the darker blue is the patients who did achieve a response, and the lighter blue is the patients who didn't. So we can see the CK response is variable. There's a trend for improvement of MMT8 in the responders. So if we take away the non-responders, we can see a general trend for improvement. And these patients, some of them are starting off at a very low level of MMT8. So to summarize the data, rese-cel was generally well tolerated across the 13 IM patients described, including 1 JIM patient, grade 1 CRS in 4 patients, no ICANS. B cells were completely and transiently depleted in the peripheral blood within 1 to 2 weeks in all of the patients. So the persistence or recurrence of autoantibodies in some of these patients may suggest the presence of CD19 negative long-lived plasma cells, which could be the primary source of pathogenic antibodies in the subset of ASAS and IMNM patients where there was a limited durability or response. So for the DM patients, there was sufficient follow-up to show 3 of them achieved response. 1 of the 2 ASAS patients achieved a moderate TIS response, and 2 of the 4 IMNM patients with sufficient follow-up achieved an immunomodulatory-free TIS response at week 24. But antibodies persisted in 3 of 6 of those patients. So based on this data, Cabaletta have initiated a pivotal cohort in DM and ASAS. So this aims to recruit 17 patients with a 16-week primary endpoint of moderate TIS off immunomodulatory treatment on no-dose steroids or low dose. Thank you for your time.
Thank you both. As you see, the QR code on the left is the one that will take you directly to our presentation in our corporate website. It's already uploaded, so you can access it there. And on the right is the link for questions. I think we have time for maybe 2 or 3 questions. The first question for Hector. From what you hear from your patients and your practice, what is the expectation for a drug-free response for when a patient receives CAR-T?
I mean, that's the big hope, that you give a potent treatment such as CAR-T, and then the patient is able to achieve a drug-free response. I think the problem that we have at the moment is there's a parallel with when biologics were introduced for patients with rheumatoid arthritis, that the patients that we're earmarking for such treatments are the ones with a very severe end of the spectrum who've proved to be very refractory. So it's something about their immune profile which suggests there may be a variable response at the moment. So it could be that once if the treatment becomes more mainstream in autoimmune disease, we'll be treating a slightly less severe phenotype with a better chance of remission, and obviously carefully choosing our patients who don't have significant damage at presentation as well.
Perfect, thank you for that. David, I think this question is more for you. Is there any data on vaccine antibody titers after receiving CAR-T therapy?
That was vaccine titers?
Yes.
Yeah. So we do have data, and this has been presented, not specifically on these patients, but in many of our patients who have received Rese-cel. And the vaccine titers, as you saw earlier with Professor Boyer's presentation, the vaccine titers are mostly maintained at the same level as they were prior to CAR-T therapy. So again, suggesting that the derivation of these antibody titers for the vaccines are coming from long-lived plasma cells, not from short-term memory B cells, which would have been ablated with the use of a CD19 CAR-T therapy.
Thank you, David. Question for you as well, David. In terms of timelines and expanding on what Hector commented on our plans for the pivotal trial for DM and ACEs, can you share a little bit about the timelines that we've set forth in terms of filing and potential availability for the product? For you?
For me?
Yes, expanding
so
on what...
This is
the
timeline for filing?
For filing.
Yeah, so as Professor Chenoy has indicated, we have an ongoing study in the cohort of dermatomyositis and antisynthetase syndrome, which is focusing on 17 patients within that patient population. What we have public stated is that we will be intending to enroll and dose the patients this year and planning to file in 2027, so within the next year and a half.
Okay, I think we have one more minute, and there are several questions that we won't get to. We will be at the booth tomorrow, Wednesday, and Thursday if you have any questions, and we're around as well, so if you need us to answer any of the questions that weren't addressed here. David, for you, can you remind us of the footprint of sites for myositis registrational trials? And the sites, the footprint of our sites.
Oh, yes, footprint for our sites. So,
yes.
So Tanya's picking on me. So the footprint for our sites, although we are focused largely in the United States because that's where we started the studies, and it crosses the entire geographical region in the States, we are also in the UK with a few sites there and looking to see if we can find additional sites in Europe to help with enrollment. In addition, for juvenile myositis, which is actually a separate group because sometimes you need to go to a children's hospital. Again, we have many sites within the US, but looking at European sites to help with enrollment for the pediatric population. Even though that is not a registrational study per se, we want to generate enough data in the pediatric population to really understand whether it will work for these patients and whether we ought to pursue a strategy to pursue an indication. Really a huge unmet medical need, and as many of you will be at the juvenile myositis session, that's an area that we really want to focus on, and if it works in kids, that will be super.
Okay. Well, thank you both for your presentations, and thank you everyone here for your time. And again, if any of the questions that you submitted, we wouldn't get to them, please feel free to drop by our booth or catch us around in the meeting. Thank you so much.
Hello. I just wanted to let you know that the MCTC session will start at 6:15 p.m.
actually, I need to open the Zoom meeting because.
Go back there.
Oh, go back there.
Yeah.
Okay.
They
okay.
are the IT people.
Okay. 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 Cool.
Hello, welcome. Buona tarde. Welcome to all of you guys. My name is Pratik Indiga. On behalf of the MCTC core group, I have the distinct pleasure of welcoming you all to this session. We really appreciate the Gcom organizers to give us some time today. We'd like to welcome all of you guys, and we would hope that you guys enjoyed the presentation today. So we're going to move a little bit quick just because we have a very ambitious agenda. We're going to do a very brief organizational update, and we really want to recognize the awardees that we have for the travel grants that have come. We want to make sure that we have some time for some very critical, interesting clinical trials updates, and then the majority of our time is going to be the panel discussion on optimizing clinical trials access and trying to set up centers, and hopefully that'll be pragmatic information for a lot of you as well. So without further ado, in terms of organizational updates, just to remember, the mission of the MCTC is really straightforward. It's to facilitate the discovery of effective treatments for IIM by coming together and really addressing the barriers to clinical trials. And we want to make sure that we have that 360-degree view with clinicians, researchers, patients, and industry, because that way all the stakeholders get to work together to really address those barriers and make sure that we're effective. And that really has resonated with a lot of people. The organization has been up and running for about a year now, and we already have over 1,000 people from across the world that are members. And that's really exciting. But what's even more exciting is that that's over 60 countries from around the world, including places that are traditionally not as well represented in clinical trials, including South America, Africa, Australia, Asia, and Europe and North America as well. And we've really tried to be reaching out and making sure that we capture the variety of subspecialists that are engaged in myositis clinical trials, including rheumatologists, but including all of the other really important subspecialties as well. We couldn't have done any of that without our sponsors, and we really appreciate their partnership and their trust and their ability to actually kind of come into myositis clinical trials, because this is an exciting time to be in the field. So thank you to our founding sponsor, Vi+Vii, as well as our platinum level sponsors, AstraZeneca, Cabaletta Bio, Novartis, Priovant, and our other gold and silver letter sponsors. So if you're interested in kind of partnering with us or other things, please come find us. But it's been a really mutually beneficial partnership so far. In terms of other things, we're excited. So thank you to T.Y. Lan and Kaushik for helping us put together our first annual report. It's a little bit of a yearbook to really kind of show some of the exciting things that we've been able to accomplish over the last year and kind of where things stand, but also to kind of set some priorities and kind of ambitions for the next year. So that'll be posted to our website pretty shortly. This past year, we've had the interstitial lung disease, the ILD interest working group, which has really brought together not just pulmonologists, but other people interested in myositis ILD. And we've kind of ran with that idea. And so we're really pleased to announce that the PRISM Partnership for Research in Skin Myositis working group is up and running under the leadership of Sweta Sadachari and Urma Khanna. And so this is really exciting, not just for dermatologists, but again, anyone that's interested in myositis clinical trials research and kind of focusing on the skin and the dermatologic manifestations of these diseases. So if you're interested, please scan the QR code. You can get more information. Along that lines, we're also excited that there's a neuromuscular working group as well, and this is to strengthen the role of neurologists in multi-center myositis clinical trials, but also to generate expert discussion and consensus on how we can expand and mentor neuromuscular neurology integration into these myositis clinical trials. And again, that's why we have kind of not only the neurologists, but everybody that's interested in this area can come together and talk about these things. This past year, we've had launched the MCTC Investigator Survey, and that's borne a lot of fruit for us. For those of you that engaged in that was actually kind of trying to get a sense in terms of who are the people that are interested in myositis clinical trials, what are their backgrounds, and that's been really helpful for trying to connect people that are interested in those clinical trials along with people that are looking for investigators. But that's also been really helpful because, as you see from some of the posters and the projects that were there, helping us do a needs assessment and understanding where are the gaps, what things can we focus on, and that's really showed some really interesting disparities and some interesting areas for us to be able to address. So the myositis clinical trials site survey is going to be launching very soon, and that one's going to end up being about the same thing. That'll help to try and connect people in terms of the sites as well as the sponsors and reduce the amount of feasibility surveys that we have. The webinar series has been off to a wonderful start, and so we have one coming up in partnership with TMA for you and your community that's really going to be focused on patients and kind of bring some of the things to them that have been here at the Gcom meeting. In terms of projects, again, we've had a lot of ambitious projects, and these have all been off to a great start. I really do want to highlight in terms of the remission and low disease activity criteria project is being submitted for ACR and EULAR sponsorship, and the evaluation and revision of kind of that FLAIR criteria is kind of up and running as a multi-group, multidisciplinary project. If you'd like to hear any more about these things, our next general body meeting is coming soon, so keep your eyes peeled, and we'll kind of go from there. So we wanted to keep the organizational updates really brief because we do want to spend some time on other things, but we really do want to recognize the MCTC Gcom Travel Grants awardees. Could I have Galina Marta and Matilda come up? So we really want to thank Shiri Carrot and Galina Marta for taking the lead in trying to make sure that we ended up having a good selection committee. Unfortunately, Shiri was unable to be here because of the conflict in the Middle East, or whatever we're calling it these days. So on behalf of her, I did want to read some of the things that she had to say. So as part of our commitment to both making sure that the myositis clinical trials in the here and present are advancing, we really want to help look towards the next generation of people that are interested and involved, and that way we can really grow the pie of myositis clinical trial sites as well as access for patients. And so along that, we're really pleased that this year we were able to announce 12 travel grants. The awardees were selected in a blinded fashion based on the strength of their Gcom accepted abstract, on their background and their potential and interest in a career in myositis, as well as kind of some of the contributions they've made to the MCTC mission of advancing myositis clinical trials. We ended up having 12 awardees, and we also made sure that we ended up trying to represent countries and areas that really had been underrepresented at global clinical myositis trials meetings in the past. And so when I announce your name, please come up to the top, and we'll start with Lakshmi Raun from India. So congratulations. Lakshmi? Hey, congratulations. And Rashid Smali from Morocco. Congratulations, Rashid. Ilyasul Abad from India.